very good friend of ours and very expert person, and he has delivered so many talks in the AICTE sponsored FDPs. The other research person will be joining us today from USA. He's happened to be our student. So both the sessions would be very stimulating, I'm sure, and they will be full of knowledge and information on the field, on the theme of this FDP. So without making any further delay, I request Tejas to introduce our first speaker, Professor Jawar sir. Tejas, you can please introduce Jawar sir to our participants.
Yes, sir. So, good evening, everyone. I'm audible clearly.
Yes, yes.
Yeah. Good evening, everyone. We are delighted to have Dr. Lakshmikant Zawar for this session in the AICTE ATAL sponsored faculty development program. Dr. Lakshmikant Zawar has been working as an associate professor and the head of the Department of Pharmaceutics at HR Patel Institute of Pharmaceutical Education and Research, Shirpur. His teaching interests include dispensing pharmacy, unit operation, hospital pharmacy, drug regulatory affairs, biopharmaceutics, pharmacokinetics, etc., etc. His primary research interests are in the field of solubility and dissolution rate enhancement, nanoparticulate drug delivery, mucoadhesive drug delivery, and polysaccharides-based drug delivery. He has guided 28 MPharm students and is currently guiding 4 PhD students and has published several research papers. He was awarded with Research Excellence Award by KBCNMU Jalgaon in 2024 for publication of research article with highest impact factor. He has also received a grant under VCRMS by KBCNMU Jalgaon worth rupees 1 lakh. He has coordinated NAC Cycle 1 for HR Patel Institute of Pharmaceutical Education and Research with the outcome of A grade. He has also coordinated one Inspire internship camp that was sponsored by the Department of Science and Technology, India. So I welcome Dr. Ella Zawar to share his expertise in nanobiotechnology with a focus on nanobiotechnology in health sciences. Sir.
Thank you very much, Tejas. Am I audible?
Yes.
Yes, yes, sir.
Yes. Yeah. So first of all, I take this opportunity to thank Dr. Sandeep Patil, who is coordinator of this ATAL FDP. I take this opportunity to thank Dr. Ravindra Patil sir and Tejas for briefly giving introduction. So let's not waste any more time. We shall begin with our topic. So I am going to share my screen. Yeah. Is the screen visible?
Yes, sir.
Yeah. All right.
Yes.
So let's begin with the talk, that is nanobiotechnology in health sciences. The contents of today's presentation are going to be introduction to nanobiotechnology, shortcomings of traditional treatment which are overcome by nanotechnology, what are the different types of nanoparticle, how do they act, what is their mechanism of action of nanoparticle, and what is the role of nanobiotechnology in early detection and diagnosis and imaging, nanoparticles for targeted disease therapy, what are the challenges and limitations of nanoparticles, and future perspective of nanobiotechnology. So introduction to nanobiotechnology. Nanotechnology is basically a science which involves the manipulation, design and development of minuscule material, which is having nanometer scale, that is range of the particle size is in between 1 to 100 nanometer, where they exert some unique characteristic which are not observed in the bulk material. For example, as we can see, gold, it is dark yellow in color, but at a nano scale level, it is dark red in color. Similarly, it will be very difficult for me to tell you what is biotechnology because it's a completely new field for me. So biotechnology uses the knowledge of biology such as genetic and cell to create and modify devices and services. And nanobiotechnology is the combination, is the convergence, is the interdisciplinary field that combines the principle of nanotechnology and biotechnology to create nanoscale tool devices and system that will interact with the biological entities, which was not possible before. So it helps to diagnose the various kind of diseases. It helps for targeted drug delivery, for regenerative medicine, and for tissue engineering. So combination by using the nanotechnology tool, the material, the devices, and by using the knowledge of biology, we are combining to make nanobiotechnology, which has various applications that we are going to see in today's talk. So before beginning with the actual different applications of nanobiotechnology, I would like to take few minutes to explore and to acknowledge the researchers and scientists who have made their contribution in the field of nanobiotechnology. So as to name the first one, it was a famous lecture of Richard Feynman in 1959, who quoted in his lecture that there is a plenty of room at the bottom. When he proposed this idea, he actually proposed this idea of manipulating individual atom and molecule. Later on, in 1975, the term nanotechnology was coined by a Japanese researcher, Norio Taniguchi. Actual development of nanotechnology and nanoparticles started when we were able to observe those subatomic level individual particle by the invention of scanning tunneling microscope. Scientists were able to visualize and manipulate the individual atom. Later on in 1985, Robert Curl and his team discovered a very important type of nanomaterial, that is fullerenes, also known as buckyballs. They are made up of carbon and they are having nanoscale range. So actually this interdisciplinary science took wings in early 2000s when the nanotechnology was merging with biotechnology, and in 2004 that resulted in the introduction of carbon nanotube.
Not audible, sir.
It's not working. Presentation has stopped. It's not audible.
Excuse me, sir. You're not audible.
Hello, excuse me, sir. You are not audible. We cannot see the slides also now.
Sir, you are not audible. Even the slides are also not visible. Please reshare the slides.
Just a second. I think there is some issues with the machine. Just a second. Hello. Sir, are you? Hello. Hello. Zover, sir.
Yeah,
Sir, your mic is off, I think.
now it is on. Huh?
Yeah. Sir, actually, you got disconnected. You can please share the screen again.
All right.
Yes.
Yeah, thank you.
Is the screen visible, Tejas, now?
Yes, sir, it is visible.
All right. Shall I continue then?
Yes, sir.
Is it okay? Shall I continue? Dear delegates, am I audible and is my screen visible?
Yes, sir.
Slide
Yes.
info is there, sir. Yeah, all right. So we were discussing what are the advantages of nanotechnology. So first of all, we already discussed about improved bioavailability. Second most important aspect that gives nanotechnology the cutting edge is targeted drug delivery. Nanoparticles can be engineered, can be manipulated, and can be attached with specific ligands, which will target the specific cells, and they will not, and hence they will minimize the damage of the healthy cell. Similarly, third advantage of nanotechnology is reduced systemic toxicity, as by targeting specific cells or tissue, we are minimizing the exposure of the healthy cells and healthy tissues. So there is a significant reduction in the systemic toxicity, which is seen with the traditional way of drug delivery. Similarly, with the advent of nanotechnology, personalized treatment is possible, which makes it very attractive method. Yeah. One more advantage of nanotechnology is controlled release. Once the nanoparticle which are attached with specific ligand reach their target site, they can remain there for a prolonged period of time, and they can release the drug in a sustained or controlled manner for a prolonged period of time. Hence, the availability of the therapeutic agent at the site of action is for a prolonged period of time. Similarly, one of the major barrier for delivery of drug to the brain is blood-brain barrier, which is a tight junction and which does not allow the passage of simple drugs which are not very lipophilic in nature. So they are being hindered for the passage through the blood-brain barrier. So which is possible now with introduction of this therapeutic agent into the nanoparticles. Then enhanced cellular uptake due to the smaller nature of the nanoparticles. They can easily be internalized by the process of endocytosis and phagocytosis. Then it also reduces the risk of first pass metabolism, as in case of traditionally administered oral or other routes of administration lead the drug delivery, introduce the drug to the liver, and there the drug undergoes extensive hepatic first pass metabolism. So due to the specific targeting by using nanoparticle, it can be avoided. And last but very important is multifunctionality. Here there is a term called as theranostic delivery. So by multifunctionality, it means the drug as well as some diagnostic agent can simultaneously be delivered in a single nanoparticulate drug delivery system, which will work in a both way for live imaging also, as well as for therapeutic or for treatment of that particular disease. So these are the advantages of using nanoparticles. Now let us see what are the different types of nanoparticles. So there are different types of nanoparticles, such as lipid nanoparticles are there. They are made up of lipid vesicles. We are going to again briefly see different kinds of nanoparticle in the coming slides. Second type of nanoparticles are metal nanoparticle. They are used for specifically for drug delivery as well as for imaging, diagnosis and imaging. Then carbon-based nanoparticles are there. Polymeric nanoparticles are also prepared. They are prepared from biocompatible and biodegradable polymers. Then self-assembled protein-based nanoparticles are also there, and extracellular vesicles are also there. So these are different types of nanoparticle. We can simply classify them as inorganic nanoparticle, which include metallic nanoparticle, ceramic, magnetic, quantum dot, organic nanoparticle made from carbon, such as polymeric nanoparticle, liposomes, dendrimer, and hybrid nanoparticle, which is a combination of inorganic as well as organic nanoparticles. So let us see in brief, because we will need to understand these different kinds of nanoparticle for their to study and to see the future application of these nanoparticle. Metallic nanoparticle, they are made up of gold, silver, or iron, and their size range is very important. They shall be below 100 nanometer, and they are basically used for drug targeting, cancer therapy, and medical imaging. Magnetic nanoparticle, again, it is a kind of metal nanoparticle which is made up of super paramagnetic iron oxide nanoparticle, and these nanoparticle have gained a lot of importance, especially in diagnosis, especially if they are conjugated with MRI imaging. So they are used for diagnosis purposes, as well as they are used for treatment such as hyperthermia treatment. Then carbon nanotube, these are cylindrical nanostructures composed of carbon atom, and they are arranged in a tube-like formation. They are also used for drug delivery and tissue engineering. Quantum dots are semiconductor nanoparticle, and they exhibit a very special characteristic, that is fluorescence. So their application is in diagnostics. Liposomes, they are lipid vesicles nanoparticle, and they can encapsulate both hydrophilic as well as lipophilic drug compounds, and they can also be modified on their surfaces for targeting to specific receptor or for targeting to specific biomarkers. Dendrimers, these are organic nanoparticle. They are highly branched, and they are made up of copolymers. They are tree-like polymers, hence they can allow us to manipulate them. We can attach different kinds of ligands on the dendrimers. They are used for delivery of drug and gene therapy. Similarly, there are certain inorganic nanoparticles which are also used, such as silica nanoparticle, which is made up of silicon dioxide, ceramic nanoparticle, which are made up of calcium phosphate and titanium dioxide, and they are having characteristic strength, stability, and biocompatibility. Polymeric nanoparticle are made up of biocompatible and biodegradable polymers, which are used for controlled and targeted drug delivery. And polymeric micelles, which are made up of amphiphilic block copolymers that can self-assemble, and they can specifically encapsulate hydrophobic drug. What are the different methods for preparation of nanoparticle? So basically there are two methods for synthesis of nanoparticle. First one is top-down method and bottom-up method. We can also classify the method of preparation of nanoparticle into, again, three more different methods can also be, there is another classification, that is physical method of nanoparticle synthesis, chemical method of nanoparticle synthesis, and biological method of nanoparticle synthesis. So in case of top-down method, the bulky material is broken down into smaller pieces and so as to create nanoparticle. Various techniques such as ball milling, that is milling, is used to prepare nanoparticle. Lithography is used. Then sonication treatment, that is sonochemical synthesis, is being used to prepare nanoparticle. And a laser beam is imparted to create nanoparticle by using laser ablation method. There are bottom-up methods. These methods involve assembly of atom and molecule to create nanoparticle. They involve various sub-methods such as chemical vapor deposition method, where chemical vapors are condensed, and these vapors are chemically reacted to create nanoparticle. Sol gel method and precipitation method are commonly used to prepare metal nanoparticle, and microemulsion method are used to prepare surfactant-based nanoparticles. What are the key characteristics which can be tailored so as to use nanoparticles for enhanced therapeutic application? So let us see the key characteristic of these nanoparticle that make them very special. So first key characteristic is size dependent property. as we know that nanoparticle size range is from 1 to 100 nanometer, and because of their such a small size, they can easily permeate through various biological membrane, and specifically without any ligand, without any binding, or without attaching any specific ligand, they can target the tumors, because tumors are often having leaky blood vessels. So they allow the accumulation of the nanoparticle in the lymphatic vessels of the tumors. So it is only due to the smaller dimensions of the nanoparticle. Also, due to their smaller size, they can easily get cleared through kidneys. Hence, they can also be used in renal disease management. Second important characteristic of nanoparticles is large surface area to volume ratio. Due to increased surface area, now reactivity could be increased. There are more sites available for interaction of the nanoparticle with biomolecules, which improve the efficacy of drug delivery system. Similarly, due to increased surface area, drug loading capacity can also be increased. Larger amount of drug could be encapsulated into these nanoparticles. Third and very important, which makes the nanoparticle very manipulative and we can do biomolecular engineering with them, that is functionalization. We can attach specific ligands such as antibody, peptide, aptamers, or some small molecules can be attached to the nanoparticle either by adsorption or by covalent linkages or by electrostatic interaction. Hence, they will target a specific biomarker, or they will target a specific cell, which will lead for the delivery, which will create the delivery of the drug to the specific type of cell. And this is very helpful in case of cancer targeting. Similarly, immunomodulation can be done by modification of the surface of the nanoparticle. The fourth important characteristic which is exhibited by nanoparticle is optical properties, which is very helpful in diagnosis and imaging, specifically surface plasmon resonance, which is a characteristic shown by metal nanoparticle, in that specifically silver and gold nanoparticle. So by making attachment of silver and gold nanoparticle, we can easily uh, view them under surface plasmon resonance. Again, second important optical property is that they exhibit optical properties based on their size. Smaller size of nanoparticle absorb the radiation, while larger particle size nanoparticles are scattering the rays. Hence, if we want the absorption of light, we will have to design our system in such a way that the particle size shall be less. And if scattering is required, then larger particles are needed to be prepared. Fifth important character is magnetic property. Again, we are nanoparticle like super paramagnetic iron oxide exhibit magnetic properties that can be harnessed for disease management, and they can be used specifically to guide the nanoparticle at specific site by using external magnetic field, as well as these nanoparticle can further be used in therapy like hyperthermia. Then they can also be, these magnetic nanoparticles can also be conjugated with contrast with advanced technologies such as MRI, which makes the facilitation of a very advanced kind of diagnostic and imaging. Then next character is thermal conductivity. Certain nanoparticles can be engineered to respond to temperature changes. Certain cancerous cells have higher temperature than the normal cells. So nanoparticles can be manipulated, can be fabricated in such a way that they will release their payload specifically at a particular temperature, and that is usually a higher temperature. So it will lead to the localized targeting of the payload for the specific targeting of specific type of cells. Seventh character is biocompatibility and biodegradability. Nanoparticles, especially organic nanoparticles, are biodegradable as well as biocompatible, while metal nanoparticle They are biocompatible, but they are a little bit tricky in biodegradability, and that is also a limitation of all these metal nanoparticle. Then controlled drug delivery application is one more application, as we have discussed. After reaching the specific target site, the nanoparticle have the ability to release the drug in a controlled manner over a period of prolonged time. Hence, availability of drug at the site of action, plenty of drug is available at the site of action. Hence, there is a tremendous reduction in the toxicity or side effect associated with the conventional therapies. Similarly, so what is the mechanism of action? What is the hidden force for the success of these nanoparticle? Take a closer look at different mechanism of action of these nanoparticle. So we can classify broadly them into three categories. That is cellular interaction and uptake mechanism, targeted drug delivery mechanism, controlled release mechanism. So first mechanism, that is cellular uptake mechanism. If the nanoparticle are prepared in very small level below 100 nanometer, specifically in the range of 1 to 100 nanometer, they are uptaken by the process called as endocytosis, while larger nanoparticles are engulfed by the macrophages by a process called as phagocytosis and pinocytosis. Again, specific targeting of nanoparticle to specific cell can be done by altering the surface charge of the nanoparticle, by altering the size of the nanoparticle, by altering the shape of the nanoparticle. Especially if we prepare certain cationic nanoparticle, they can easily target the particular cell uptake will be very easy, as the cell membrane is having negative charge. The second point which I would like to emphasize over here is surface interaction. Nanoparticle can interact with cellular receptors which are involved in immune responses, and they can either be internalized, that is engulfed. Hence, nanoparticle can deliver the drug inside the cell, or they can modulate the cell signaling by specifically binding to specific receptors. Again, we can modify the surface of the nanoparticle so as to target a specific type of cells. For example, liposomes can be coated with PEG, hence so that they cannot be detected by RES, that is reticuloendothelial system. Third important point is how can we do the targeting of nanoparticles. So there are two mechanisms of targeting of nanoparticle. First one is active targeting and second one is passive targeting. In case of active targeting, as we can see from this diagram, this is an example of liposome. It's a lipid vesicle which contains bilayer, and we can manipulate the surface of this liposome by several different ligands, such as aptamers can be attached to the surface of the liposome. They can be attached by using adsorption, they can be attached by covalent linkages, they can be attached by some cationic and anionic, that is polyelectrolyte complex interaction. So various methods are there for the ligand attachment to the surfaces of nanoparticles. Then proteins could be attached, peptide could be attached, antibodies could be attached, and this attachment will lead to the specific targeting ability. It will give the specific targeting ability to these nanoparticle. As in case of example of certain antibodies can be attached to the nanoparticle, which will specifically target the HER2 breast cancer cell, and which will keep aside the healthy cells. In case of passive targeting, no special ligands are attached. Only due to the size of the nanoparticle, they are attracted towards the leaky vasculature of the tumors or inflamed tissue, and in this way they get concentrated at that particular site. And through the mechanism of fenestration, that is small doors or openings or small openings are there in these leaky blood vessels, through them they can easily concentrate at that particular site. Again, there are certain other mechanisms are also associated with the nanoparticle, that we can design nanoparticle in a such a way that they will respond to certain specific pH. As in case of liposomes, they react to the acidic environment, and liposomes, there is a conformational changes happening in the liposome that will release the drug specifically at acidic pH. Similarly, thermosensitive nanoparticles can be prepared when they are exposed to heat, such as hyperthermia treatment. They will release the drug at that particular site. Then we can also design nanoparticle which are enzyme sensitive. Ligand has to be attached, so that biomarker has to be attached so that it will bind to the specific enzyme and it will target that specific enzyme. And the last mechanism of all these nanoparticle is they release the drug in a sustained and controlled manner by these two mechanisms. That is, diffusion controlled release is taking place, where slow release of the nanoparticle over a prolonged period of time takes place, and a matrix controlled release is taking place, where polymeric matrix or a coating is done on the surface of the nanoparticle. Now let us see in short, because it's a very vast field and a lot of work is being done. Let us see what are the applications of this nanobiotechnology in early detection and imaging. So the first application, and that has revolutionized the current diagnostic therapy, is quantum dot. The problem with the conventional therapy is that we are able to diagnose a particular disease after manifestation of symptoms. So once the disease has progressed at a higher level, then symptoms occurs, and after manifestation of symptom, the disease is being diagnosed, some tests are written. But with the help of nanotechnology, even before the manifestation of symptom, the disease can be diagnosed, and that could be done with the help of various tools such as quantum dots. So traditional diagnostic rely on antibodies are bounded to specific dyes, and they produce fluorescence, and that fluorescence is of not accuracy and specificity. So that is a disadvantage of traditional method of diagnosis, specifically in case of fluorescent imaging. While if same antibody or same ligand, if it is attached to quantum dot, which are made up of inorganic nanoparticle, then they will show very bright fluorescence, because they have certain advantages such as broad absorption spectrum. They can absorb the light starting from UV until the visible range, that is from 200 to 800 nanometer. While very important characteristic advantage is that they emit the light at a very specific narrow emission range, and single wavelength could also be excited, and hence the overall there is an enhancement in detection of that particular target cell or of that particular biomarker could be possible with the help of quantum dots. Second is protein chips or microarrays. Protein chips they immobilize the protein on microslides, and then certain target moieties, they bind to this protein molecule and hence we can diagnose or detect a particular disease. But immobilization of protein on microslides have a limited surface area. This technique could be further enhanced by creating a surface of gold or a similar metallic nanoparticle, which will give enhanced surface area. And on those nanoparticles, then proteins are immobilized, so they can be detected by using resonance spectroscopy. So immobilization method and surface functionality can be done. Two products have been developed. One is deep pen nanolithography by Nano Ink, and inkjet technology have been developed by Agilent Inc. Third is DNA target probe. Here, what is done is that metal nanoparticles are combined with the single strand of DNA, which can combine in biological fluid, in biological sample. If there is a pathogen or if there is a genetic material, then that can bind with this strand of DNA. Hence, diagnosis, single molecule detection is possible with the help of carbon nanotube. Then genetic detection, segment of DNA segment labeled with gold nanoparticle can detect specific genetic sequence, and they are very useful in detection of prostate cancer, breast cancer, and anthrax, and so many more diseases. Sparse cell detection is also possible with the help of nanotechnology. What are sparse cells? So sparse cells are the defective cells, like HIV infected T cells or cancer cell or fetal cell. So they have certain different characteristic than the normal cells, such as they have different surface charges, they have certain anomalies, they have certain defects or deformation in their cell. So that could be diagnosed, that could be detected with the help of gold plated nano or metal organic framework nanoparticles can be used for detection of these sparse cell, as well as they can be separated. These sparse cells can be also separated by using metal nanoparticles. Then what are the applications of nanoparticles in disease therapy? We'll look in short one by one what are the different applications of nanoparticles in various disease therapy. First application is nanoparticle in gene therapy. The current challenges of gene therapy is that gene mutation reverting to the original form can take place. Then immune reaction can be triggered by viral vector, because currently viral vectors are being used for delivery of gene. This could be overcome by using nanocarrier based gene delivery technique, where inorganic nanocarrier ranging from 50 to 500 nanometers effectively encapsulate the gene. Then these nanoparticles can further be modified with certain kinds of ligands so that they will specifically target certain specific target cells, and then they will release their payload. They will release their gene in the intracellular level by the process of different mechanism we already seen, by the process of endocytosis, by the process of phagocytosis, so or by interaction with the receptor at the cell surface. The gene delivery is possible, although it is still in the preliminary stages. Then one more advantage of delivering gene through nanoparticle is that they are non-immunogenic. They do not produce any immunogenic response which could be produced in case of viral vector. Another application of nanoparticles is in biomolecular engineering. So nanoparticles can be engineered in a variety of way. We can attach different kinds of biomolecule, as it could be seen from this diagram. We can attach different kinds of antibodies could be attached, fluorescent proteins could be attached, peptides could be attached, enzymes could be attached, antibody fragments could be attached. The traditional biomolecular engineering is time consuming and expensive, while this nano biomolecular engineering using nanoparticles could be cost effective and more efficient. Similarly, they will allow the synthesis, this biomolecular engineering could be done on solid substrate, which is not possible with the conventional biomolecular engineering, which require the solution-based method. The major application which is being explored for nanoparticle is in cancer treatment. Just a second, please. So nanoparticles are used in cancer treatment for targeted drug delivery, such as doxorubicin drug. It is encapsulated in liposomes to target specific type of cancer cell. Again, different kinds of cancer cell could be targeted by engineering with the different kinds of ligand. Nanoparticles, nanobiotechnology could enhance imaging characteristic in combination with MRI, CT scan, and fluorescent. Then they could also be used for photothermal therapy. That is light infrared, external exposure of infrared light will lead to the accumulation of the gold nanoparticle at that specific site, and hence destruction of cancer cell is possible. Similarly, gene therapy and immunotherapy is possible. Genetic material can be delivered to boost immune boosting agents could be delivered to cancer cell, aiding in targeted gene therapy. And nanoparticle can overcome the chemotherapy resistance by encapsulating drug in ways that will bypass the resistance mechanism. Then these are the few examples. This Doxil product is already in the market. It contains doxorubicin. It's an chemotherapeutic agent used in cancer. It's a broad spectrum anti-cancer agent, and it is encapsulated in liposome. It is a form of nanoparticle, and it is further coated with pegylated. That is, pegylated liposomes are created. PG layer is so that they could not be detected by RES. Similarly, cisplatin, it's a chemotherapeutic agent. By encapsulating it in a nanoparticulate drug delivery system, scientists were able to reduce its toxicity to the healthy human cell, and they increase in the therapeutic index and effective against tumor. Methotrexate is again one kind of chemotherapeutic agent which is used for the treatment of leukemia, and it is being investigated and it is being delivered through carbon nanotube to reduce the systemic toxicity as well as to improve its pharmacokinetic, because it is having poor solubility. Similarly, there are other drugs such as paclitaxel, which is also a BCS class 2 drug, which is very poorly soluble. So it is being formulated in the form of albumin nanoparticle. The paclitaxel is encapsulated in albumin-bound nanoparticle, which improve its availability, bioavailability, and hence therapeutic activity is improved. So similar kind of drugs are encapsulated in various kind of nanoparticle, such as gemcitabine and vinca alkaloids are encapsulated so as to improve their therapeutic efficacy as well as so as to reduce their toxicity. Then nanoparticle in antiviral prevention and treatment. So first of all, the recently launched vaccine, that is BioNTech and Moderna for COVID-19. It's a nanoparticle based vaccine which contain mRNA, which is encapsulated in lipid nanoparticle, and it contains the mRNA encapsulated in lipid nanoparticle. If given through parenteral route, it can produce immune responses. Similar is the case with the Novavax vaccine, which contains, instead of mRNA, it contains the spike protein, which are encapsulated in again same lipid nanoparticles, and they result in the triggering of the immune response. Then certain antiviral agents such as remdesivir are being developed in the form of nanoparticles to treat SARS-CoV-2 virus. Then ritonavir is also being prepared in the form of lipid-based nanoparticle. So for the improved delivery in the therapy of HIV. The major mechanism of action for delivery of this drug through nanoparticle, what advantages do they bring is there are three important things are happening. First is due to the encapsulation of this drug into the nanoparticle, they disrupt the viral cell membrane, causing leakage and cell death. Second mechanism is that if the drug is being encapsulated into the metal nanoparticle, metal ions are created, and these metal ion disrupt the metabolism, leading to oxidative stress. And also these nanoparticles generate reactive oxygen species, causing oxidative damage to the lipid protein and DNA. So this is the mechanism through which they can also overcome. These are also the mechanism for overcoming the resistance of the chemotherapeutic agent, as well as in case of delivery of drug to certain kind of bacteria. These are the mechanism through which we can overcome the resistance to the conventional delivery of the viral agent as well as bacterial antibacterial therapeutic drug substances. Then in case of antibacterial treatment, again, same mechanisms are there. They disrupt the cell membrane of the bacteria. Then they create reactive oxygen species, and third thing is that they are disrupting, they cause the damage. Again, a very important application of these nanoparticles, especially the silver nanoparticle. Previously, silver sulfadiazine was directly used to treat wound care, but nowadays silver nanoparticle, in fact, the products are also marketed and we are frequently using them in case of minor burns, ulcers, or chronic wound. So silver nanoparticle are encapsulated in gel form. They are available and they are very effective in comparison to the conventional silver sulfadiazine ointments or cream. Then hospital associated infection, specifically silver nanoparticles are used to coat the medical devices and surfaces, hence to reduce the risk of infection caused by antibiotic resistance bacteria like MRSA, that is Staphylococci and E. coli. Then nanoparticulate drug delivery system is in Alzheimer's and Parkinson's treatment. Alzheimer's disease is caused, it causes memory loss and confusion due to brain cell degeneration. So Alzheimer's drugs, that is anti-amyloid drugs, could be delivered through nanoparticles to combat the plaques which are formed in the brain. And also insulin delivery is also under investigation to deliver to the brain. The main challenge of the conventional drug delivery to the brain is that the conventional therapy is very limited amount of drug is able to cross the blood-brain barrier. And by encapsulating the drug in the nanoparticulate drug delivery system or nanoparticles, this barrier can be overcome. Again, this barrier can be overcome by changing the route of administration, such as through nasal route. These nanoparticle can be delivered to target the brain cells or for targeting into the brain various kinds of brain diseases such as Alzheimer's and Parkinson. Genes could also be delivered. Then diagnostic imaging could be done with the help of quantum dot and iron oxide nanoparticle in combination with MRI and PET scan. One specific example of delivery of anti-amyloid drug that reduces the plaque is PAMAM, that is polyamidoamide. It's a kind of dendrimer which encapsulate the drug and it could be delivered to the brain and it reduces the aggregates or plaques which are formed in the brain. In case of Parkinson's treatment, Parkinson's disease is a neurodegenerative disorder that affect the movement, causing tremor, stiffness, and balance problem due to the loss of dopamine. So again, conventional oral therapy later on leads to increase in the dose of the drug, and due to continuous increase in the dose of the drug, it could lead to side effects such as dyskinesias. So by encapsulating these L-DOPA and various kinds of genes and neuroprotective agent in the nanoparticles, it could be very effective way of treating Parkinson's disease. In case of diabetes, devices are being developed which will continuously monitor the level of glucose. Wearable kind of devices are being developed which will continuously monitor the level of glucose. Hence, frequent finger stick test is avoided, and they will also systematically will allow us to adjust the dose of the insulin. So specifically in case of diabetes, in case of detection of glucose is the main focus for the this nanoparticle. Various kinds of quantum dots are developed which emit fluorescent signals upon the, and where we can determine these are glucose sensitive quantum dot which can produce specific signal for blood sugar measurement. Similarly, colorimetric tests, these are POC, point of contact. Immediately there is a change in the color if there is a change in the concentration of the glucose level. These kind of instruments and these kind of devices are being developed. In case of cardiac diseases, specifically statins are encapsulated into the nanoparticle so that they can target to specific sites where the clots or where the atherosclerosis like condition is being developed, and thereby targeting there, they can reduce the plaque formation. Then a complete diagnosis, imaging and diagnosis could be done for the various cardiac disease by using gold nanoparticles. In orthopedic applications, the nanomaterials like nanotube, nanopolymer, ceramic nanocomposites, and carbon nanofiber, they improve the mineral deposition, especially calcium-rich compound, enhancing the bone quality. Then they are also being used for effectively attachment of the implants, so that the interaction between the bone and the implant and retention of the implant will be there. Then methotrexate, it's a chemotherapeutic agent as well as it's a drug which is used in treatment of rheumatoid arthritis. We can specifically target this drug by encapsulating it in the nano particulate drug delivery system to the site of rheumatoid arthritis. Then in case of dental therapy, nanoparticle are being developed such as hydroxyapatite. They are helping to repair the microscopic cracks in the teeth. Then targeted delivery of drug in case of dental therapy is possible, specifically at the gum or the teeth site. Then certain materials are being developed such as silica in the nano form, which can cover with which various kinds of implant and crowns can be made, and a layer of enamel which will be long lasting is being developed. Then antibacterial and anesthetic agent are also delivered using nanoparticles. Now we are coming to the last segment of this today's talk, that is challenges and limitations of nanoparticle. One of the major challenge of nanoparticle is that of toxicity, specifically the metal nanoparticle like silver and gold nanoparticle. If given in high concentration, they could be lead to toxicity. Then regulatory approval and safety is again one of the second challenge. Stringent regulations are required, and extensive safety testing is being done so before approving this kind of dosage form. And in fact, FDA is also refining the guidelines for the development of a nanoparticle. Then one more major challenge is the production and scalability. Nanoparticles on a small scale can be manufactured, but for a large scale manufacturing of nanoparticle with uniform size, shape, and functionality, it's a challenge. Then controlled release and targeting. Nanoparticles after development sometimes they could release their payload immediately. So that's a limitation of nanoparticles. So sophisticated engineering is required so as to ensure that precise targeting as well as a controlled release of the drug could be done. Then environmental, we are yet to explore what is the effect of these different kinds of nanoparticle on the health of the human, on the health of the animals, on the health overall, on our biodiversity, on what is their impact on the environment is yet to be explored. Then what are the future perspective of nanobiotechnology? So nanobiotechnological advancement nowadays, actually lot of work is being done by encapsulating herbal compounds, that is herbal remedy. The material or individual component from herbs are being used to treat or are being tested to treat various kinds of diseases, and they are being used to treat specifically cancer. So various kinds of individual phytochemicals are being used, and the future direction is going to be the combination of the herbal individual constituent and nanoparticle for the treatment of various kinds of disease. Then nanoparticle, although few formulations are there, but still a lot of advancement is possible in cancer treatment, specifically with the in vivo trials are for efficacy and safety. Then non-invasive delivery. Research is increasingly being focused on non-invasive delivery methods, especially for ocular and cardiovascular disease, as well as reproductive medicines are being tried to deliver through non-invasive way to treat that disease in a non-invasive manner. And the most important future perspective of nanoparticle is the personalized medicine in diagnostic. So in future, probably tailored nanoparticulate or targeted drug delivery system could be developed, which will be suitable for individual patient. So these are the references I have used while preparing this presentation. And lastly, I would like to say that just like the sunrise brings a new day, nanobiotechnology offers a bright future with new possibilities to improve human health.
Thank you, sir. Thank you, sir. I request participants, if you have any queries, you can raise. Yes, please, from participant side.
Hello,
Yes.
sir. Good evening, sir. So
Arun,
this is Vaishali from Nasik.
Arun Palanisamy.
Hello. Good evening,
Yes,
sir.
Dr. Arun.
Yeah, this is Dr. Nalini. Hello, sir. Can you hear me? Hello, sir. Can
Yes,
you hear
yes,
me?
ma'am. Please, please.
Sir, I would like to know how you make the silver nanoparticles.
Yes, Dr. Arun, you have raised your hand. You please unmute yourself and
No, sir,
ask
it
your
is
query.
Sandeep, sir.
Yes.
It is Dr. Nalini. She is asking something.
Okay.
Dr. Nalini, please go ahead.
Yes, sir. Good evening, sir.
Good evening.
I would like to know how you deliver the nanoparticles, the methodology that could be discussed here. Silver nanoparticles, sir.
Yeah. So silver nanoparticles are nowadays being used specifically for wound care. So silver in nanoparticulate form, then final formulation is being done in the form of gel, which can be directly applied at the surface of the wound. So basically, it depends upon where is the target site. Where do we want to target such kind of nanoparticle? In case of silver, nanoparticle are predominantly used for wound care. So simple gel formulation are being prepared and they are being delivered through direct application.
Actually, in silver nanoparticles, many green synthesis, they are doing green synthesized nanoparticles, right?
Yeah.
So in that case, how about gel can withstand that?
Actually, there are various methods are there for the synthesis, as it's again, it will become a very long topic for synthesis of silver nanoparticle. But once these nanoparticle are prepared in the form of suspension, they are quantified, they are tested, and then simply, so specifically talking about the delivery for the wound site, a gelling agent such as carbapol or polaxamer in a required concentration of 1 or 2% could be added into those suspension which are purified, and then it could be delivered at the site of the wound. Or
Okay, sir.
simple, in some cases, these nanoparticles are lyophilized and they are made into the powder form. And these powder formed are again mixed with some diluent. And this powder formulation is sprayed on the inanimate objects, such as, say, for example, in case of various devices which are being used for hospital.
Okay. Thank you very much, sir.
Can I ask a question, sir? Vaishali from Nasik. Hello. Hello.
Yes, anyone else?
So can you hear me?
Yes, ma'am.
Audible.
So I'm Mrs. Vaishali Chue from Nasik.
Yes, Dr. Vaishali, ma'am.
Sir, when you're administrating my nanoparticles, what are the side effects of these?
Yeah, again, I would like to tell, we are actually discuss the side effect. The major side effect of nanoparticle is that we need to ensure that they are being engineered in such a way that they will reach specific target site. If something goes wrong in the engineering of the nanoparticle, then we will unable to deliver them at specific site. That is one. Second thing is that if they are not formulated or prepared properly, then they could not deliver the drug in a controlled manner. So burst release will happen, and that is a worst case scenario. Like suddenly all of the drug will be released. Third point is that specifically in case of a metal nanoparticle, they could lead to toxicity. Specifically, as they are accumulating in the kidneys, they could lead to renal toxicity. So these are the limitations of nanoparticles.
Thank you.
Hello. Can you hear
Good
me,
evening,
sir?
sir. Am I audible, sir?
Yes.
Sir, am I audible?
Yes.
Hello.
Yes, sir, you are audible. You please go ahead.
Yeah, thank you, sir. Thank you, sir. Sir, it was actually provides a number of information regarding nanotechnology. Actually,
Yeah,
I'm having
actually, it's such
maybe
a
I'm not sure
vast
about
topic, and I have tried to cover as many things as possible so as to give the glimpse of how
many
can we
information, sir. Many information.
how can we combine? Because I came to know that the most of the attendees are from biotechnology, microbiology field, and I belong to pharmacy field, and we work on preparation of nanoparticles. So I thought that to compile various applications of nanobiotechnology, so some idea or some information I could probably give to all the delegates.
Yes, sir. Yes, sir. Thank you so much, sir. My question is, like, is there any ethical concerns are there when people are producing this particular nanoparticles, sir? Ethical concerns, sir.
Ethical permissions.
Ethical concerns, sir, in production of nanoparticles, sir.
Yeah, please go ahead. Please complete, and then I will try to answer your question.
Yeah. Is there any ethical concerns there when we are producing the nanoparticles, sir?
Okay. Again, while testing, yeah, of course, we'll have to go through different preclinical, definitely while development of any pharmaceutical or any dosage form, there are ethical concerns are there. And for that, during development stages, proper permissions has to be taken from the regulatory agencies. So yes, there are ethical concerns, but by taking proper permissions from regulatory agencies, they could be developed and they could be checked in case of preclinical trials could be conducted, as well as clinical trials could be conducted. As in case of the delivery of mRNA vaccine, with that is Moderna and Pfizer. So it is that product is already, we already use that product. So yes, there are ethical concerns, but they need to be addressed through a proper channel. I hope I have answered your query.
Yes, sir. Thank you so much, sir. Thank you, sir.
Hello, sir. Can you hear me?
Yeah, please.
Hello. This is Dr. Avinash from Nasik. I would like to raise one question regarding the biological nanoparticle synthesis. I think when I go through your presentation, you have given a lot of stress on the chemical and the physical synthesis of nanoparticles. We microbiologists are actively engaged in the biosynthesis of nanoparticles from variety of microorganisms. I would like to ask regarding the quality and the yield of nanoparticles obtained from these chemical, physical, and biological methods. Which one is better?
Actually, it's a little bit difficult question. But yes, I also mentioned during my presentation that apart from top down method and bottom up method, there are three methods. We can classify the methods in different ways. One way of classification is top down method and bottom up method. Another way of classification is physical method, chemical method, and biological method. And yes, it's an upcoming topic for generation of nanoparticle using cells or using microorganism. It's a very upcoming issue, but frankly speaking, I have not worked on the methods for generation of nanoparticle using microorganisms. So I will be unable to answer which is a better method,
Hey, regarding quality point of view, suppose silver nanoparticles I have synthesized by chemical method and biological method. Regarding the quality and efficacy point of view,
so
which one is better?
Quality point of view, we need to check the as the raw material is going to be the biological source, we need to check what kind of impurities are being also imbibed, or what kind of impurities are going to be carried. While in case of synthetic process, they are actually well established processes are there, synthetic processes, while biological processes are still undergoing development. So a lot of work is needed to be done.
Okay, sir. Thank you. Thank you very much.
Thank you.
Yes.
Good evening, sir. Am I audible, sir?
Yes, please.
Myself, Dr. Bhanumati Singh. Sir, we have been actively involved in the green synthesis of the gold nanoparticles with the help of peels, fruit peels. But my question is, I just want to know that there are some sunscreen products which have been utilizing zinc oxide and titanium oxide nanoparticles. And they have been used by the sportsmen and other people who are the swimmers on their skin for to avoid this UV radiation. So, what should be the exact size of these nanoparticles that should not affect the skin also? Because I had studied that there must be certain size of these nanoparticles that we should consider that should not deteriorate the texture of the skin.
Specifically for skin, I would like to actually I would require to go through specifically, because that is an area where I have not worked. So right now I will be unable to tell you exact what kind of size will be required. But usually, if you speak metal nanoparticle, then the size of the metal nanoparticle is below 100 nanometers.
Okay. Thank you, sir.
Thank you very much.
Yes. Next one.
Good evening, sir.
Yeah, please go ahead.
Sir, it was very informative lecture. My question to you is, how successful are biologically synthesized nanoparticles?
Means the method utilized for biologically synthesized nanoparticle.
The method means if we are using biological systems for synthesizing nanoparticles. So which system works best, and is it really successful as compared to the chemically synthesized nanoparticles?
As previously, actually, we discussed with one of the delegate that uh, here we cannot say that this method is the best method or this method will give. Again, we'll have to go through various methods, and similar nanoparticles could be prepared by using chemical methods and biological methods. And actually, a lot of research is needed to be done so as to comparatively evaluate which will be a better method.
Okay, but sir, I wanted to know your views on this, your expertise on nanoparticles. Based on that, what you would like to comment on success of the nanoparticles? Which one should perform better?
Actually, we are preparing at my lab. We are preparing using some physical and chemical method. We are yet to explore biological methods for the preparation of nanoparticles. We are actually at my lab. We are preparing some protein nanoparticles, and they are being prepared by precipitation method. That is bottom-up method is being used for preparation of nanoparticle, as well as sometimes to prepare liposomes, we are using thin film hydration method, and micro emulsion methods are being used. So we are using chemical methods for synthesis of nanoparticle.
Okay, okay. And sir, one small point is there. Chitosan nanoparticles. I wanted to know briefly about chitosan nanoparticles from you, sir.
It's actually a wonder polymer. There are thousands of articles are available for the synthesis of chitosan nanoparticle. Basically, it's a cationic polymer. So interaction and attachment of ligand is possible with the chitosan nanoparticle. It's a very versatile polymer which can further be modified for attachment of different kinds of ligands for targeting specific type of cells. So a lot of work is being done actually on the preparation of chitosan nanoparticle. Lot of modified form of chitosans are being utilized for synthesis of nanoparticle. Very well established methods and procedures are available for the preparation of chitosan based nanoparticles.
Okay, okay. So thank you so much for answering all the questions. This was Afreen Khan from Nasik. Actually, I am working I am a PhD scholar. So my topic is based on nanoparticles only. So if possible, I would connect with you for further guidance, sir.
Yeah, sure. We can cooperate. Yeah, sure.
Yes, sir. Thank you so much, sir. I am obliged. Thank you.
Thank you very much.
Yes. Anyone from audience again? If no one is there, I request Ravi sir to please finish.
Thank you very much, sir, for very interesting and stimulating session. In fact, it is the topic of very general interest, and so many participants are interested, to do the work based on nanobiotechnology. And I hope they will be keep in touch with you in the near future, and I'm sure that you will certainly guide them. So if there is no any question, I conclude and I thank Dr. Jawar for being with us. Thank you very much, sir.
Thank you very much. And now I think that I would also like to collaborate with Micro and Biotechnology Department. And I'm looking forward to go into details about the biological methods for nanoparticle preparation, because it is an upcoming topic and I have got 2-3 questions on the same line. So surely I'm looking forward to go through the details of the biological methods for synthesis.
Certainly, sir. Certainly, we are already working on it. And if anybody is having any, if they need any assistance at the primary level, we will certainly open for that. All right. We are doing some work on biosynthesis of nanoparticles using plant extracts and as well as microorganisms, and we are exploring nanoparticles for different biological activities. So if you have any issue, you can certainly write to us, or even you can phone call to us.
Again, thank you very much for giving me the opportunity to deliver my talk in this FDP. Thanks a lot. Thank you very
Thank you
much.
very much, sir. So, Sandeep sir, are we waiting for our next speaker?
Yes, we are ready with next speaker, Dr. Rahul.
So, are you going to start or shall I go ahead?
Yes. I request Dr. Rahul to please unmute yourself.
Hello, good evening. Can you hear me?
Yes, Rahul sir.
Thank you, sir.
Yes, he's there, sir.
So, good evening, Rahul.
Good evening, sir.
What's the time there exactly now?
It is exactly 7 a.m. in the morning.
So sorry, we troubled your morning.
No. Actually, I'm happy that you scheduled the presentation at the evening in India,
Right.
so that I can manage presenting in the morning. Otherwise, I have to wake up in the late night and deliver the talk.
Right. So shall we start?
Yeah, sure. Should I share?
Just a minute. We'll introduce you and then you can go ahead. All right.
Sure.
So, ladies and gentlemen, I take pride, to let you know that we have with us our best alumni, Dr. Rahul, and he is working in US right now as a postdoctoral fellow at a very wonderful institution. And he is our product, learned in a very small place and reached to the great height today. And I'm very happy that he accepted our invitation and he is ready with his talk. So without making any further delay, I request Tejas to please introduce Rahul sir to our audience. So Tejas, it's over to you.
So I'm going to introduce Rahul. So I really feel immense pleasure in welcoming our speaker for the next session, Dr. Rahul Suryavanshi. Dr. Rahul is notably the alumni of the Department of Microbiology and Biotechnology of R.C. Patel Arts, Commerce and Science College, Shirpur. He is currently working as investigator tenure track neurovirology unit chief at the Laboratory of Neurological Infections and Immunity in National Institute of Health called NIH in Hamilton, Montana, USA. Before this, Rahul has worked as a scientific program leader at the Department of Virology and Immunology at Gladstone Institute, San Francisco. He has completed his PhD from KBC North Maharashtra University, Jalgaon, and postdoctorate from the Department of Microbiology and Immunology at University of Illinois at Chicago. He has received an award of excellence by Gladstone Institute of Virology and Immunology in the year of 2021. He has also received an award of excellence by the Department of Ophthalmology, again from University of Illinois, Chicago in the year of 2020. In 2017, he has got Erasmus Mundus Fellowship, which is very prestigious, that is awarded by the European Union to carry out research work at the University of Perpignan, France, where he has one of the only two candidates from India. He has near about 73 Scopus publications, where two are in Nature. He has also published 3 book chapters and has filed 2 patents. Today, he'll be sharing his expertise in virology with a focus topic in vivo studies to understand virus pathogenesis. So I conclude with this introduction, and this is over to you, Rahul.
Yeah, thank you very much, Dr. Ravi Patil sir and Dr. Sandeep Patil sir. It is indeed a pleasure to meet you and deliver my talk for this very important event for development of faculty throughout the India. And thank you for such a nice introduction. Now I would like to share my content.
It is there at the upper right corner.
Yeah, I saw it. Can you see my slides now?
Yes. You please maximize your screen.
Yeah. Now it's okay?
Yeah, it's okay.
Great. Yeah, before I start, I would like to tell that if you have any question, you can stop me right in the middle and you can ask me any doubt. So am an experimental virologist who studies viral pathogenesis using different animal models. And for today, I have opted for giving you an overview of the use of animals in understanding the viral pathogenesis. Sorry. Yeah, but before I talk about my research, I would like to give you overview of my research journey so far. As Dr. Ravi Patel sir and Dr. Sandeep Patel sir told, I have completed my master's in biotechnology from R.C. Patel College in Shirpur in 2008. And meanwhile, I worked at National AIDS Research Institute for 3 years, where I worked on HIV, and there I was motivated to work on viruses. Uh, but after completing 3 years of research over there, I got an opportunity to enroll for PhD at KBC North Maharashtra University in the School of Life Sciences under potential guidance of Dr. Satish Patil sir, where I worked on pigment producing bacteria, mostly on proteogene producing organism called Serratia marcescens. And I worked and collaborated internationally to understand the biological effects or to understand the drug potential of the pigment proteogene. Later, in 2016, I got an opportunity to work as a postdoc in the University of Perpignan in France. Here, I received a fellowship from a European Union called Erasmus Mundus, but my virology work resumed at a postdoc at University of Illinois Chicago. Here in Dr. Deepak Shukla's lab, I worked on herpes simplex virus 1, which majorly causes infections in the cornea in US and mostly to the orofacial regions in India, but is also known for its viral pathogenesis in the brain. Then early 2021, when the SARS-CoV-2 pandemic was high and SARS-CoV-2 Delta variant was grooming around the world, almost every one of us has been impacted by the virus. So I decided to join classroom institutes who had a biosafety level 3 facility suitable for work on SARS-CoV-2. And meanwhile, I was also hit very hard by the virus and it affected my life uh, at the family level. So I decided to continue working throughout my life on the emerging pathogens. And therefore, I uh, decided to join a National Institutes of Health, where I am currently working as a chief of neurovirology unit, and I have my own lab where we work on long-term impacts of SARS-CoV-2 and other emerging infections, specifically respiratory viral infections. So in today's presentation, this is the index. We would briefly go through the mice models that we can use to study SARS-CoV-2. I will give you a brief account of using the animal models in SARS-CoV-2 virus transmission, pathogenesis, behavioral studies or neurological manifestations of virus. Then I would give you overview of studies on the microbiome and how we can study the immune response using animal and even humans. And I haven't added this point, but how we can screen the different pharmacological candidates in SARS-CoV-2 infected animal models is something that I will cover in this same topic. And then I will give you a detailed overview of my studies on the long COVID. As we know, SARS-CoV-2 is almost an endemic now. So it is causing only mild infections, not life-threatening infections. But almost 10% of infected individuals are facing long-term side effects of the SARS-CoV-2 infections. And I will cover the neurological and cardiovascular side effects of SARS-CoV-2 in long-term using animal models. So starting with the animal models that we can use on SARS-CoV-2. So my purpose of this presentation is to make you aware of how we can make use of animal models for particularly one disease. And that's why I have selected topic of SARS-CoV-2 infections. But I also have slides for how we can study the herpes simplex virus infection using animal models. But I'm not sure if the time will be enough to present those slides as well. So I'll stick to SARS-CoV-2 for now. So for working on SARS-CoV-2, as you can all understand, the SARS-CoV-2, because it transmits through air, we cannot work in normal biosafety cabinets or laminar air flows that we normally use in laboratories. It needs a biosafety level 3 facility. We call it when we use animals in the BSL-3 facility, it is called as animal biosafety level 3 facility. And these facilities are very rare. Even in US, there are only few facilities for animal work in the BSL-3. Even in San Francisco, where I was working for these studies, there were only 3 labs having capability of including animals in their biosafety level 3 facility. So you can understand the level of stringency or the level of security that is needed to perform these operations in research. So in our animal biosafety level 3 facility, I work with 5 animal models. You can see 4 of these are mice and 1 is Syrian hamster. For the mice, it can be a normal wild type mice, or it can be a transgenic mice or virus induced mice. So first we'll talk about K18 human ACE2 mice. As some of you may know that SARS-CoV-2 uses ACE2 as a receptor for infecting human cells. But this receptor was not specific in animal models. Therefore, some of the earlier variants of SARS-CoV-2 like Wuhan, Washington, Delta, they were not able to infect mice. But in order to study the disease, we need the mice model. And therefore, we used a transgenic mice, which is capable of expressing human ACE2 onto their cell surface so that we can facilitate the entry of virus in the cell. And that is the animal model which is used for studying the severe effects of SARS-CoV-2 disease. Then the second mice model is wild-type mice. This is the animal model which allows the infection of many other variants like SARS-CoV-2 beta variant, gamma variant, beta variant, then all the Omicron variants that we know, that is BA1, BA2, BA5, BQ1.1, or XBB, XBB 1.5, JN1, etc. So it is suitable for all other virus and variant infections of SARS-CoV-2. And the characteristic of this mice model is it causes mild infection. So the virus doesn't kill these animals like it happens in 98% of individuals with SARS-CoV-2. Whereas K18 animal, they are all killed by Washington, Wuhan, or Delta variant or Beta variant, but not by other Omicron variants. The next animal model that we can use is Black 6, that is wild type animal, but transduced with adenoviral vector, which is capable of expressing human ACE2 in the lungs. This animal model has a specific ability to express human ACE2 in the lungs only, so that we can restrain the infection in the lung area only, and the infection doesn't go to brain or any other parts of the virus of the body of the animal, so that we can particularly study the effect of the SARS-CoV-2 infection in the lung. Then I would like to talk about diversity outbreed animal model. So diversity outbreed animal is genetically complex animal model. I have recently started working on this. There are few reports on the other genetically complex animal models, but this is the model that is being studied by only my lab currently, and we are getting very exciting data. And I think we will be able to delineate some of the interesting facts about SARS-CoV-2, particularly the reasons behind why SARS-CoV-2 infections were particularly more virulent in some individuals. Like some individuals were getting severely infected, while others were getting mildly infected or moderately infected. So we can understand the genetic factors behind the disease severity of the SARS-CoV-2 using this animal model. And the Syrian hamsters, which some of you might know, they look like mice, but they are not mice. They are at least 3 to 4 times bigger than the mice. They do not have longer tail. They're very sweet, and they are pets in other countries. And they are the best animal models for studying the virus transmission that I will show you in a few minutes. Because virus transmits from air, so infected individual, human, can transmit the virus through air to another individual, but somehow mice models lack this ability. So they cannot transmit the virus through aerosols, but hamsters can, and therefore we have to use hamsters for virus transmission studies. Before jumping into how we can make use of mice for studying the SARS-CoV-2, I would like to give you an overview of the current status of SARS-CoV-2, which, like at this point of time today, although we know that SARS-CoV-2 has been endemic, but SARS-CoV-2 is still spreading throughout the world. Although it is causing very mild infections, but it is still spreading. And I should say we are protected because we are vaccinated. And the vaccination is also contributed by the herd immunity that is protecting us from severe side effects of newer variants of SARS-CoV-2. But I must say that SARS-CoV-2 is currently gaining changes on differential immune landscape. Like some of us are naive. They have never been infected, they have never been vaccinated, and the virus can infect them. Although this particular population is very low in number. Then there are a group of individuals who are convalescent for SARS-CoV-2 infection, who never vaccinated but were infected by the virus. So they have the natural immunity against the virus infection. They are called convalescent. There are individuals who have vaccine-mediated immunity, meaning that they took the vaccines but were never infected with SARS-CoV-2. And this is one of the second most effective immunity against SARS-CoV-2. Then there are individuals with hybrid immunity, meaning those individuals who were vaccinated as well as infected by the SARS-CoV-2. They have hybrid kind of immunity by natural infection and artificial immunization. And this is one of the best immunity, which is broad spectrum, as well as it gives a high titer of the antibody, which I would explain in a few minutes. Then there are individuals, specifically old individuals, who lose the antibody titer or immunity very soon after infection or vaccination. So we call it as a waning down of immunity. And these humans are also now susceptible for newer infection by newer variants. And almost all of us now have least humoral immunity or antibody mediated immunity because it's been more than 2 years for us to take to take any vaccine. Although in developed countries the booster shots are still ongoing, people are taking vaccination every year to maintain the antiviral immunity. but I'm not sure in developing countries or underdeveloped countries, the vaccinations are not ongoing. So the people are more susceptible for newer variants in future. And there are individuals who are falling sick because of virus infection, and they're taking medications. So it is one of the important development in the field that there are now medicines to treat the infection. So we don't need to worry much about the virus, but it is not able to prevent the long-term side effects of the SARS-CoV-2 infection, which we will talk in the later part of the presentation. So on all these immune backgrounds, the virus is adapting itself to give rise to newer variants. And some of these variants sustain for some time before fading away, and some of them continue modifying it themselves. So as I said before, now the variants are causing mild infections due to vaccination and herd immunity. But there are some hospitalizations in elderly individuals or immunocompromised individuals. But the main reason that we need to worry about is the long COVID, which appears after 1 year of infection, 6 months to 1 year after infection, which include neurological consequences. Just to tell you a brief example, some people feel lazy, uh, dizziness. Some people say that, oh, the name of this person was on tip of my tongue, but I forgot. So people are experiencing cognitive deficits. So they are losing their memory. We have been observing increment in the cardiovascular deficits or malfunctions. There are blood clot problems in individuals because the SARS-CoV-2 antigen is still lingering around in their blood, which causes or gives rise to blood clots, which we will see in the later part of the presentation. There has been increment in the heart attacks, and we will also explore how we can address or study the effect of virus infection in the lung on the heart of the animal models, which may correlate in humans. And there are impacts of immune dysregulation, which we will also touch base on. So before I begin again, a brief overview of what I do. I study the virus infection, different variants of virus, first in cell culture, and then I make use of animal models for studying their virus transmission, pathogenesis, drug screening, and long COVID. So we'll begin with virus transmission. As you know, the virus is transmitted through air with the aerosols, which can be inhaled by potential host or normal individual and can gain the infection. Now here we make use of Syrian hamsters. And how do we do it? Is we infect this donor hamster with SARS-CoV-2 Delta variant or any other type of variant actually of SARS-CoV-2. We make this as viral suspension and infect the animal through the droplets in the nose. So that through nose, it goes to the lung and infects the animal. Now this animal is infected and have the potential to infect other animals. Now, in order to study the virus transmission on day 2 of infection, so this is day 0 when we infect the animal, and day 2 is 2 days after infection, the Syrian hamsters which are infected are co-housed in a cage with the recipient for 1 hour. And then the recipient hamsters are isolated from the cage, and they are caged independently and euthanized or sacrificed at day 2 post co-housing, that is day 4. And then we euthanize the recipients, sorry, recipients, and harvest their lungs and nasal tissues to understand the virus through plaque assay. So here the clear zone in the blue background represents one plaque, and we call it as one infectious particle. And we analyze the virus titer based on the plaques. that we get. So the more the plaques, the more the virus in the lung of the animal. This is the actual experiment where you can see the cage, which is divided through a mesh, which permits the air to pass through, but not let the animal to touch themselves. So on this side, we have donor animal, and on this side we have the recipient animal. So one donor animal and two recipient animal, and the air can pass through the mesh and infect the recipients. So this was about the virus transmission studies. Now, how we can study the pathogenesis of SARS-CoV-2 variants in mice. So pathogenesis, uh, for this particular part, I will focus on replication of the virus, how we can study the replication of the virus in the lungs or the brain. So here we make use of wild type or K18 human ACE2 mice. They are infected at day 0, and at day 2, day 4, or day 7 post-infection, we euthanize subset of animals, like 5 animals from each group on each time point. And we harvest their lungs and the brain regions to perform plaque assay to understand the virus replication. We also monitor their temperature, their body weight, their survival, and we check the virus through histology as well. So this was the data where we infected mice with 3 strains of the SARS-CoV-2, that is Washington, Delta, and Omicron. And the animals were euthanized at day 2, day 4, and day 7. These studies are published in Nature in 2022. And you can refer to the paper. This is the first figure of the paper, where we were able to see that Washington and Delta are causing changes in the body temperature, that the animals are becoming hypothermic. Their temperature is decreasing as the days goes on. And while Omicron, which is comparatively less pathogenic, it has comparatively normal phenotype in the animal. So it is less impacted by the virus infection. When we see this figure C, there has been a steep decrease in the body weight because the animal is infected, it is feeling uneasy, it is having virus in the brain, specifically in the ACE2, human ACE2 K18 animal. They're losing the body weight and also reaching to the humane endpoint. They're not eating well, they're looking lethargic. As you can see, the Washington strain infected animal is hunched and it is looking morbid. Also, the Delta infected animal is also lethargic. It is moving very slowly. While the Omicron infected animal, they are behaving normal. So both Delta and Washington, they are almost dead by day 7 to day 8 post-infection, but the green line here shows Omicron. It is perfectly fine. And this is why we did not experience lethal phenotypes because of Omicron infection in humans too. This is the histopathology that we can study, where we take out the lung, we section the lung, and we stain the viral protein with green label. So all this green part on the blue background you can see is the viral protein. So, and this is a section of the lung. As you can see at day 2, day 4, and day 7 post-infection, for Washington, there has been a lot of infection at day 2, day 4, but it reduces by day 7. Same case is with Delta variant, but with Omicron, the infection is far less. And you can see the the Omicron causes like small spots, whereas here you can easily see it. So Washington is causing spread to the neighboring cells, but Delta is causing much more bigger zones of infection, as it can be seen through this plaque assay as well. So the plaques with the Delta virus are bigger, whereas with the Omicron virus for same amount of same days of infection, they are causing very minor dots. I feel myself that this is one of the primary reason behind less pathogenicity of the Omicron, because it has less ability to spread from one cell to other cell through the wall, and so that it cannot escape from the human immune system. Whereas Delta variant uh, passes from one cell to the other cell through the wall of the cells. So it doesn't go outside and neutralized by the immune cells, but it infects the cell in a group, and that is how it causes bigger zones of infection and makes more damage to the lungs, and that's why it has more lethal infections in general. So I won't talk about the new variants. As you know, there have been immense number of variants arising every day. But I also use this mice model to understand the pathogenicity of the newer variants at day 2 and day 4 post-infection. So here I use wild-type mice and wash the SARS-CoV-2 beta variant, which is pre-Omicron variant. Like it was arised in 2021, 2022, from 19 to 22, but later it was taken over by Omicron variants. So initially the Omicron variants also had less ability to infect the animal, but as we can see, the developments of the virus until XBB 1.5, there has been consistent improvement in the ability of the virus to infect the animal. So, but we are still protected. Although the Omicron regained the ability to cause more pathogenicity or virulence, we are still protected because we have been vaccinated, and we also have developed the herd immunity, which is protecting us from the development of or modifications in the SARS-CoV-2 variants. But you never know. You have, and therefore scientists need to keep on monitoring the infections, keep watch on the newer mutations happening in the SARS-CoV-2 variants to save the world from next pandemic, and that is what we are doing. Now, in addition to the mice models, we also developed a technique to study the virus infection in directly human lungs. So we received some lung tissues from humans, donors, from human donors, and those tissues were converted into single cell. And with the help of some growth factors, we were able to make a small chunk of the lung. That is a piece of lung tissue, which is a combination of different cell types like ciliated cells, club cells, basal cells, and goblet cells. And we feel that this is also a better model to study the effect of the drug or virus pathogenesis for SARS-CoV-2. As you can see, you must have heard about the term called organ culture, right? So this is a kind of the same where we have developed the lung tissue in the Petri dish. Here you can see this is an organoid, and it has some beating cells or ciliated cells which are moving on the organoid surface. These are the cells which produce mucus, which comes in our mouth. And we were so happy to see this, that we can culture from the single cell, we can develop an organ, and we can study it further for SARS-CoV-2 pathogenesis or drug screening. Also, we make use of 82 cells from the human lungs, the primary cells, which are also effective to study the air-liquid interface, um, which mimics human lungs in our body. The third application of using animal models is to study the gut microbiome. As most of us know that gut microbiota or organisms in our gut play a very important role in our digestion or overall pathophysiology or physiology of our body. Many times when people have stomach problems like loose motion, doctors prescribe us to eat curd or to get probiotics. because they are very important to maintain the good health of our gut. It has also been observed that gut microflora is responsible for our mood swings. It also controls our nervous system for some reasons. And therefore, it is very important to maintain good gut health, to have rich microflora, which consists of the beneficial microbes. But the viral infections can upset our gut, and they can affect the colonization of bacteria in our gut. And we studied the mice we studied the gut microbiota using the mice model, where again the mice were infected with SARS-CoV-2, different variants, that is Washington, Delta, and Omicron. And we performed 16 sRNA sequencing and um, checked with for different families of bacteria, um, and we observed that uh, compared to uninfected, the Washington or Delta infected animals showed lower presence of varico microbiota, uh, and a little increase in the bacteriodota. But the impact was more severe for the mild infectious variant, that is Omicron. So Omicron, which causes actually least effect or least disease severity, it has even higher impact on decreasing the gut microbes, which also help in our immune system. So we are still working on this to understand the effect of reduction in particular family of microbes because of virus infection and how it impacts uh, different physiological processes in the body of animal. These studies were published in the journal called MBio in 2023. Now, the most important and the topic of my current interest is about the behavior. You know that our behavior is a natural predictor of our neurological health. How we are thinking is better predicted by our behavior. And we can study the behavior of the animal and can understand if the virus is impacting the nervous system of the animals. I will give more information about this experiment in the later part of my long COVID slides, but here I want to show you how I did setup for the behavior assembly in the biosafety cabinet inside the biosafety level 3 facility. This was more challenging to set up the assembly inside the biosafety cabinet in the BSL3 to monitor the movements of the animal. We have the camera located outside the biosafety cabinet. The camera can check the reflection of the animals present in these two chambers, one in each, through the mirror which is slanted below the chamber. And this can accurately track the movements of the mice. I will share a small video. Can you see the video? Can you see the mice moving?
Yes, sir.
Yes, sir.
Okay, cool.
Yes, sir.
So this is a machine learning approach. Yes, this is a machine learning approach where we can track different body parts of the mice, and we can also track the movements of the animal inside the chamber. This is very interesting and very important technique, which can give us knowledge about how the animal is behaving in the chamber after virus infection. Is it causing any memory loss? It is causing any behavioral problems, anxiety, fear conditioning, etc., etc. We can track everything with the use of behavior studies. And this is the first study developed by us, and we are currently finishing the story, and we will soon publish in Good Journal. Then we also used animals to study immunity. But these slides, I'm going to present the data of humans. We recruited the human population to understand the humoral immunity. Some of you might know what humoral immunity is. So we have specifically two types of immunities, like innate and adaptive. And in adaptive immunity, we have humoral immunity and cell-mediated immunity. What is T-cell mediated immunity? So antibody-mediated immunity or humoral immunity is something that gives us quick protection against SARS-CoV-2 after like 7 to 8 days of virus infection. Basically, what happens is when virus infects us or our cells, there are some cells called antigen-presenting cells like macrophages, neutrophils, etc. They they digest the virus and then they present the viral antigens on the cell surface. And those antigens, by the way, this knowledge was given to me by Dr. Ravi Patil sir when I was studying MSc Biotechnology in RC Patel.
Yes, I
So it is.
remember. I remember Rahul.
Yeah. This was, and he referred us to a book called QB, and I was such a fan of that book. He really introduced me to Stryer for molecular
Right.
biology and QB. Those are the two most interesting books which really motivated me to do science. So antigen presenting cells, they can
Rahul, just sorry to interrupt
yeah.
you.
No
These
problem.
are these icons you are using from BioRender or somewhere else?
Yes, BioRender.
BioRender, na?
Yeah.
Yeah. Thank you.
Yeah. No problem.
Actually, we are also trying to use it, but it's very costly, na?
Okay. Okay.
But it looks nice.
Yeah.
Okay, please go ahead. Go ahead.
So NIH has a free resource.
Right.
I will
I
share
understand.
with you. Yeah. So I will share the link.
It will be really helpful, if it is possible.
Sure thing. Yeah.
Sure.
Yeah. They have recently launched it and it is freely available. Anybody can use it. And it's as good as BioRender. So the B cells can recognize the antigens presented by antigen presenting cells, and they can produce specific antibodies which will specifically bind to the SARS-CoV-2 virus or antigen and neutralize the virus. And we can study this using an assay called virus neutralization assay. Here, what you do is you take the blood from the animal or human, which is either vaccinated or infected. You isolate the serum, and you can make different dilutions of the serum and incubate it with same amount of virus particles, like 50 virus plaque forming units, let's say, and then perform plaque assay. And as you can see, with higher serum, like lower dilutions of the serum, you can see clearance of the virus infection. Whereas when you dilute the serum more, so with higher serum dilutions, you get less and less neutralization of the virus, so more and more plaques. This is the easiest way to understand how we can study the humoral immunity against any virus, basically. I learned this essay in my second postdoc at University of Illinois, Chicago, against herpes simplex virus 1. So this data is published in Nature, and we were able to understand that. Excuse me, I'm hearing
Please
some
put off
background
your mic.
noise. Please put yourself muted. So there were humans which were unvaccinated but were infected by Omicron variant. There were humans which were unvaccinated but infected by Delta variant. We compared them with the individuals who were vaccinated and Omicron infected and vaccinated and Delta infected. Likewise, we check their humoral immunity. So here, this axis is called is representing neutralization titer at 50%, and the value over each bar, like 1452 here, represents the NT50 value. So higher the value is the higher neutralizing potential or higher antibody titer against that particular virus. And here we check the titer against Washington variant, Alpha variant, Beta variant, Delta variant, and Omicron variant to understand the use of vaccination and which type of immunity actually gives us more neutralization against the virus. So every dot represents one human being. So these all humans were enrolled in this study to understand the humoral immune responses in those individuals. So when the humans were not vaccinated but Omicron infected, we saw highest neutralization of Omicron in these individuals. Um, uh, but those individuals have least ability to neutralize other variants. Meaning, if you are naturally infected by Omicron, it will confer immunity only against Omicron, at least humoral immunity. There may be cell-mediated immunity or T-cell mediated immunity, but humoral immunity is conferred only against Omicron. Same thing with Delta. The highest immunity was conferred against Delta and to some level by against Washington, but not against Omicron. Meaning, if you are getting natural infection and you are not vaccinated, you have very limited or less specific immunity against different variants. But let us say if you are vaccinated plus Omicron infected, you have the highest immunity against Omicron as well as other variants. Meaning, vaccination is always helpful for you to strengthen your immunity in the context of infection as well. Same result we observed with vaccination and Delta infection, that the immunity was highest against pre-Omicron as well as Omicron variants, and therefore we estimated that hybrid immunity is one of the best measure of protection against the disease, which can give us broad spectrum um, protection against the virus. But that doesn't mean that you should get infected with virus even if you are vaccinated. But vaccination will decrease the disease severity in your body, and it will actually strengthen your immunity if you are getting infected. But don't expose yourself to infection, because even vaccinated individuals are experiencing long COVID symptoms, and we don't want that. But vaccination itself can protect you from different infections. Uh, so now I will turn my focus on the detailed studies. Here, I will also talk about the drug screening experiments. But before that, the SARS-CoV-2 and herpes simplex virus, they are the two viruses I have worked on, but today I will be talking only about SARS-CoV-2. SARS-CoV-2, although we know that it primarily causes infection in the human lung, but it also impacts human brain and human heart and other like immune system, respiratory system, circulatory system, and we will give specific emphasis on the neurological manifestation, that is its impact on the brain. We have the slides for herpes simplex virus 1, which is also neurotropic virus, causes encephalitis in some individuals. By the way, herpes simplex virus, which causes blisters around our mouth area, many of us do experience these blisters after a fever. We call in Marathi as a sinu barlai, means there is some viral infection after our immunity is down after fever. And this also causes infection in the brain, which I'm currently studying, and its impact on the development of Alzheimer, dementia, cognitive dysfunctions. There are many things that we can study for the post-acute or long-term effects of the SARS-CoV-2. So we'll begin with SARS-CoV-2 again. I have studied the pathogenesis. As you know, we have published in Nature for Washington, Delta, and Omicron variants that I showed data for. I have studied other variant pathogenesis as well, which is still unpublished. Yeah, this is the data where I showed a brief slide about this, that Omicrons are regaining the pathogenesis behavior. But again, a mutation in the XBB1.5 can drop down the ability of the virus. So this is keep on going. Like some of the variants, they are gaining higher ability to infect animals, while another mutation in those variants are decreasing the pathogenesis. So this will keep on going. So we have to keep dealing with that. Now, this is very interesting area. That is humoral immunity. I showed you how we can measure the titer of the antibody, antiviral antibodies in different individuals with hybrid immunity, artificial immunity, and convalescent immunity. And here, very interesting data that I have observed is that the red line here represents hybrid immunity. The blue line here represents artificial immunity conferred by vaccination, and the yellow line here represents um, convalescent immunity or the immunity conferred by natural infection. So what I have observed at different days post-infection, like at day 28 to 29, the hybrid immunity or artificial immunity reaches to its uh, peak level. That means highest antibody titer are observed in that particular time. But there is difference where when we compare to the natural immunity, it peaks up at a very later time point. This is very important basic understanding that is not there in the book, QB or in any other immunology book, to my understanding. And I want to pursue this study further to understand how the vaccine-mediated immunity is effective to get a higher titer and quicker build-up of the antibody response, and why the natural infection causes late increase in the antibody titer, and it is very specific. So this is something that is ongoing. Then I have also studied the cell-mediated immunity. I will show a brief slide of this cell-mediated immunity work that we are doing currently. Here we have animals divided in 4 groups. The animals that are uninfected, that is we call as a mock. The animals that have been vaccinated, the animals that have been BA1 infected, like Omicron infected, and the animals who are vaccinated as well as Omicron infected. We gave them a vaccination dose in particular groups, 2 doses of the mRNA vaccine, and then we harvested their serum samples at day 30 post vaccination or infection, and at 45 days and 66 days we harvested their tissues. Um, sorry, we harvested the tissues at 66 days post infection, and particularly, I mean, it's a lot more slides, but I'm presenting only one slide here. We have observed one specific marker present on the immune cells, that is CXCR4. So CXCR4 is a cell receptor present on many immune cells, which causes trafficking of the immune cells from primary secondary lymphoid tissues to the infected areas, to the lungs in this case. And we have particularly observed that natural infection causes increment in this expression of CXCR4. This is also observed in people having long COVID, that CXCR4 expression was higher even 1 year after infection, meaning they may have dysregulated immunity contributed by the expression of this particular factor. And we are currently doing animal studies to understand the impact of CXCR4 expression on the behavior, on the neurological deficit, and immune dysregulation using the anti-CXCR4 antibody, which we gave to the animals for until 21 days every other day after infection. So the study is in progress, will be published soon in Good Journal. After this, I would like to give you a brief overview of in vivo studies, how we use the animal models to screen the drugs. These studies are also published in good journals and have been patented for a couple of drugs, which may come in the market sometime next year. As I have told about the organoids, we can make use of organoids. We can infect the organoids with the virus and then treat them with different concentration of the drug. Here we have a drug candidate called AVI4206. This was just yesterday, it was published in the journal eLife. You can check it. It clearly shows that at higher concentration of the drug, there is less virus replication. And same drug was used to treat the SARS-CoV-2 infected animals, where SARS-CoV-2 was infected by Washington isolate, um, human isolate, and the drug treatment was given BID, that is twice a day, through IP at 100 mg per kg. The ABI 4206 treatment showed less replication of the virus in the lungs of the animal at 4 and 7 days post-infection, whereas the mutant for that particular protein, that is macrodomain, was incapable of replicating at a higher level, which served as a positive control in our experiment. And we proposed this as a concept. We developed this as a drug target and then developed drug against that target to help reduce the severity of infection for future variants of SARS-CoV-2. Then we have another drug called MPRO inhibitor. So MPRO is a protein of the virus. It is called as the main protease of the virus. We can target the viral proteins particularly to inhibit its replication. And this was one of the most effective drug that we have observed so far. No, actually there is one more drug, that is 4773, which is even better than this. So at day 2, day 4, and day 7 post-infection, in comparison to positive control, that is nirmatrelvir, AVI 4516 showed better inhibition of the virus. As you can see, it is consistently protecting the virus at different time points. At day 7, it is actually clearing the virus infection. And at different concentrations of 4516, we see dose-dependent effect on the virus inhibition at day 2 post-infection by AVI 4516. We can also study this using histopathology. As you can see, the brown color represents the virus replication. It is highest with the vehicle control at day 2, day 4, and day 7 post-infection. With nirmatrelvir, it is little, but with 4516, it is the least, which we can show. And we can also study the inflammation, if anything, with H and E staining of the animal tissues or lung tissues from the mice. So this was about the drug screening. We can do preclinical studies using animal models to screen the drugs against SARS-CoV-2. Now I would turn this paper has recently been accepted in Science Advances journal. Now I would turn my focus on long COVID. So what is long COVID? Now this is the detailed study. What is long COVID? Long COVID is a long-term effect of the virus infection on human body. It can cause abnormality in the brain functioning, normal functioning of the brain, normal functioning of the lungs. Some people feel that they are tired, their oxygen levels is low, hypoxia. Some of them are experiencing increment in the heart attacks, which is pertinent to the circulatory system, and some are experiencing immune dysregulation. And we particularly focus here on the brain and circulatory system. Even in the brain, as published in Nature Medicine in the year 2022, people are experiencing many different phenotypes. They can experience cerebrovascular disorders, mental health disorders, neurologic or related disorders, then some movement or extra pyramidal and movement disorders, or even migraine or something. But our particular focus was cognitive disorders, the problems related to the memory of the animals after SARS-CoV-2 infection. So we studied the memory deficits after SARS-CoV-2 infection with the behavioral studies, and then we studied the virus-host interaction and immune activation in the brain to understand how the behavioral studies were impacted by SARS-CoV-2. For this, we collaborated with Palop Lab in the Glasgow Institute, where again I'm showing the same setup, where sorry, I think I'm able to play this video again because I have selected the marker. Okay, now I can play. So we can track the movements of animal in the chamber. This technique was developed by Mackenzie Mathis. They are the husband and wife. They developed this technology, and now it is revolutionizing the behavioral studies across the globe. This is a very new technique, and we make use of machine learning approach or artificial intelligence to study how the behavior changes in different situations and how we can predict the neurological consequences of any virus infection or any disease using this tracking of the movements of animal. This can also be done on humans or any other wild animal. So what it actually does. So the camera or software tracks the body movements, so the pattern of body movements. And these patterns, they may be running, they may be backward running, forward running, stationary rearing, or grooming, uh, rear grooming, back grooming, etc., etc. These motifs are compiled by the machine learning approach. We call it as a variational animal motion embedding WAME analysis. And with this WAME analysis, we can plot the graphs to understand the behavior of the animal. Now, particularly for memory, we did observe. So what was done? These set of 10 animals were added in the chamber, and the idea was to see the chamber. So when the animal is first added for the first time in the chamber, its natural behavior is to explore the chamber, right? It will check for the food, it will check for the security, it will check for any risk, danger. Uh, so it will move around the chamber a lot to explore it. But let us say if we are putting the same animal in the same chamber after 21 days or 1 month after this first presence, this time it should remember that, oh, there is nothing in this chamber, so I should not waste my energy to exploring it. So it should remember, it should recall its memory, right? So what we did, we put the animal at -1 days pre-infection, we infected them at day 0, and after 21 days of infection, we put them again. So here you can see the distance traveled by mock or SARS-CoV-2 infected animal. At day 0, there is no difference between the two groups. But at day 21, the red line, that is virus infected animal, are still moving a lot compared to the mock infected animal, meaning that the infected animal are still exploring the chamber. Exploring the chamber, meaning that they don't recall the chamber. They don't recall the chamber means they are having memory problems. That's what is predicted, and these studies have been confirmed by other researchers as well. So one thing is clear, that animals infected with SARS-CoV-2 show some memory deficit, and now our next challenge was how we can study it further. So to find out the reasons behind this central nervous system manifestations of COVID-19, we checked back on the references, and several researchers from NIH figured out from the patient samples or brain samples of humans infected by SARS-CoV-2 that there is a presence of viral antigens in the brain of the dead individuals which died because of COVID-19. And these, when there is a presence of viral antigen, there is an inflammation. Inflammation, meaning immunity, presence of immunity is there, like microglia, some T cells, some B cells, etc. So this inflammation, we call immune system as an army of our body, which can protect us from the infection. But this army, if it is staying in your home for a long time, it can also cause inconvenient situation. Suppose they are colonizing in Jalgaon or in city for a long time, it actually makes your life difficult. And same thing happens in our body. If the inflammation is there for more time, it actually makes you uncomfortable and diseased, and it can infect your memory, impact your memory. Now, the next question was, which type of immune cells are causing problems to the cognitive or causing problems to the memory or causing memory deficit, cognitive deficits. There have been some papers published which hold microglia activation responsible for the memory deficits or dementia. So microglia are type of innate immune responses which are antigen presenting cells which also capture the antigen or virus and digest it. And my next target was to see if the microglia activation happens during the acute phase of SARS-CoV-2 infection, that is at day 2. At day 2 post-infection. I'm sorry, I did it at day 3 post-infection, which is still very immediate after virus infection. So what I did, I infected wild-type animals with SARS-CoV-2 beta variant. At day 3 post-infection, I harvest their brain. And harvesting their brain, we took the cross-section of the brain slice and checked for and stained the slide for microglia protein, that is IBA1. And as you can see here, so mock or uninfected animals show very less density of the microglia, but SARS-CoV-2 infected animal shows higher density of microglial activation. As we quantified it, the different regions of the brain like cortex and hippocampus are showing more activation of microglia compared to uninfected animal. And you can also see the shape of these microglia as the microglia in the uninfected animal. Their central portion is smaller and the extended bodies, fibers, filaments are longer. Whereas in activated stage, the microglia, the centromere, it becomes larger and the extended bodies becomes shorter. Sorry. Can you still see my slides?
Yes, sir.
Yes,
Okay.
sir.
Okay.
Yes, sir.
Thank you. So as I said, when the microglia is at resting phase, it is the centromere is smaller and then the filamentous bodies are longer, whereas in the active state, the centromere is larger and the filamentous bodies get shorter. But our next question was, what viral or host factors activate these microglia? And it has been observed the presence of spike protein, viral spike protein in the brain. So we accounted for presence of viral spike protein in the brain, and then we also notified that there have been increment in the blood clotting. You remember in the beginning there was debate that there have been blood clots in the infected animal, in infected humans, and the blood clotting factor in humans called fibrinogen. We thought that it might be responsible for activation of microglia because it has already been reported. So we considered 3 factors, that is SARS-CoV-2's spike protein, fibrinogen, and microglia. They may be interacting with each other. And it was further supported by the evidence rationale by studies in NIH, where infected humans who died because of SARS-CoV-2 infection, they showed presence of fibrinogen in their brain and activated microglia in their brain. So we thought that we should study the co-localization of fibrinogen with SARS-CoV-2 spike protein. And therefore, again, we did a collaboration with Akaso Lab and Green Lab at Gladstone and repeated the same experiment where animals were infected with SARS-CoV-2 beta, and at 3 days post-infection, we checked their lungs this time. We stained the viral protein with green antibody, fibrinogen with red antibody, and as you know, green plus red, it produces yellow. And we were able to observe that wherever there was a co-localization or binding of spike and fibrin, there was yellow color in the tissue. And when we pull down this fibrinogen, with the immunoblotting, we were also able to Western blot the spike protein in these cell extracts or tissue extracts. The important thing about this is we also observed the presence of reactive oxygen species. Now, ROS, we study in biochemistry. The ROS is the activator, or it is produced by microglia or macrophages when they are active. And we did observe that we simply brought blood from the blood bank and we increased the number of the spike protein uh, and in those blood from blood bank, and observed that the more the spike, the more is the blood clot formation, meaning that it was very clear that the spike protein from virus can cause the blood clot formation. And when we put this spike fibrin complex on bone marrow derived macrophages isolated from bone marrow, um, we were able to observe consistent increase in the reactive oxygen species with respect to increment in the spike protein in these cells. Meaning, the more the spike protein, the more the activation of macrophages. And macrophages in rest of the body, they are called macrophages, but when the macrophages are in the brain, they are called microglia. Therefore, we wanted to study if the virus infection through the lung causes any impact to the brain, and does it cause activation of microglia? And we were able to do so. We saw that compared to uninfected wild type animal, SARS-CoV-2 infected wild type animals showed fibrinogen deposits in the brain. And in the same region or in the nearby region, we were also able to identify the microglia. So IBA1 is a protein which accounts for activated microglia. And as you can see, the microglia are very predominant in the SARS-CoV-2 infected region. But then we asked a question: what if there are no fibrinogen in the animal? And we have the fibrinogen knockout animal. So as in US is highly resourceful for research resources. We have so many transgenic animals which we can make use to study the functions of these important complexes or proteins. So we made use of fibrinogen knockout animal. And in fibrinogen knockout animal, when we infected them with SARS-CoV-2 and compared them with wild type animal, we were able to see less presence of microglia activation. This is again CD8, CD68, IBA1. These are the combination protein for microglia activation. And as you can see, a lot more staining in the microglia population compared to fibrinogen knockout animals. Now, it was very clear to us that microglia are being activated by spike fibrinogen. But can we study any cure for the disease? And for that, we aimed to focus on the antibody called 5B8. So 5B8 is one of the antibody which has been published before by our collaborators, which particularly binds to fibrinogen. The fibrinogen region, which is immunomodulatory. So I will explain this with the graphics. So here, as we know that the spike binds to fibrin. Now, if we add 5B8 antibody to this complex, it reduces the binding of spike to the fibrin, and it also reduces the reactive oxygen species formation. So here, when we added more and more antibody to the complex, there was a dissociation of spike with fibrin, and there was also decrease in the reactive oxygen species formation. As you can see, this is actually reducing the activation of microglia because reactive oxygen species act as a marker for activated microglia. Now, our next goal was to understand if this is having any therapeutic or prophylactic potential in the hippocampus of the animal. And I'm glad to see that 5-8 treatment reduces the activation of microglia and put it back to the resting phase, whereas control antibody is still showing strong activation of the microglia. As quantified with both prophylactic, that is before infection, and after infection, if we treat the animals with 5-8 antibody, we can see a reduced activation of microglia. So with this, we think that it can also act as a drug to cure the patients who are suffering from long COVID for neurological disorders. We also took account of RNA sequencing, RNA-seq for a different type 1 interferon signaling genes. This is just giving account of the highest inflammation observed with control antibody, whereas the inflammation was reduced overall with the 5-8 treatment. This finding has been published in Nature in 2024. So in summary of this part, we can say that the neural manifestations of long COVID are associated with a memory deficit, and the spike fibrinogen binding stimulate the activation of microglia, which may cause memory deficit, and therapeutic intervention by antifibrin 5-8 antibody can reduce the binding of spike and fibrin and reduce the activation of microglia in the brain and can act as a cure for neurological manifestations of long COVID. And by the way, 5-8 antibody is also being used for Alzheimer's disease already. Now, this is the last part of presentation. I think it is the cardiovascular manifestation of long COVID. This data has never been shown to anybody else, but you are the first people to see this. But I'm actively working on this and will soon publish a paper out of it. So as we know that cardiovascular diseases have been on the rise after SARS-CoV-2 infection, but not all the humans experience cardiovascular manifestations. Few of them do. So I studied different animal models to see if I can see the virus replication in the heart of the animal, and did observe that the diversity outbred animal, which is a genetically complex animal, is the only animal which shows the virus in the heart, while the wild type or K18 human history animal, they do not show. And this is why this research is still lacking behind in this area, because people don't know which animal model is the best animal model to study the virus infection in the heart. But fortunately, I was able to understand that diversity outbred animals can be used for this studies. Now, why the diversity outbred animal are showing the virus infection in the heart? As you can see, the diversity outbred is a combination of 8 different animals. Like in humans, we are breed from like different population, right? So more complex genetics contributes to more variation in your genetic factors, and they contribute to your disease severity. Like some individuals experience high virus replication, some experience moderate virus replication, some experience very mild virus replication. And this phenotype is only showed by the diversity outbred mice. Whereas wild type or K18 mice, they show high or severe infection, which is not the case in humans. We experience differential disease severity. And with this, this is the section of the heart where I was able to see the fibrosis in the heart, which is also one of the reasons behind the heart attack. And one more notifiable thing was there was increment in the troponin level. So whenever a person is having heart attack, doctors or physicians, they do check one important test, that is troponin levels in your blood. And if the troponin level is high, meaning that your cardiomyocytes are being damaged, and they release the troponin in the serum, which is an indicator of heart disease. And with the diversity of the animal, I was able to see that some of the animal, not all, are showing increment in the troponin levels, whereas wild type animals with different strains of SARS-CoV-2 do not show such an increment, although few animals did. So my goal is to understand which host factors in these animals are contributing to the disease severity, particularly in the heart in these animals, and which factors which we should consider before beginning the treatment, predicting the disease severity, potential disease severity of SARS-CoV-2 infection. I won't go through this, but my long-term plan is to continue working on the immunological and neurodegenerative sequelae of SARS-CoV-2, and also work on the impact of SARS-CoV-2 infection on the memory deficit with not many behavioral assays. I won't go in this, but similar to lung organoids, I also work with the brain organoids. But my long-term goal is to make use of diversity orbit animal to perform some detailed phenotyping and make use of organoids to understand the host factors that are contributing to disease severity, identify those host factors, and develop the drug molecules which may help us to improve the disease outcome by targeting novel drug targets with new chemical entities in future. And for this, I have established a lab at National Institutes of Health, where my main goal is to do basic as well as translational research. My focus is on to understanding the pathways, investigating the pathways of cell death and immune activation and their interaction with the host factors, to study the acute and long-term or post-acute neuronal consequences associated with virus infections, and to develop the therapeutic interventions to resolve the neuropathologies caused by the viruses. With this, I would like to acknowledge my colleagues at Gladstone Institutes who helped me to execute these experiments, my supervisor, Melanie Ott, and my friends and my research collaborators who immensely helped me for executing this study. I didn't talk about herpes simplex virus, where I was principally supported by Chandrasekhar Patil, who is my colleague, friend, and we work collaboratively on many research projects, and we will continue doing so. He has helped me a lot. And of course, to the funding agencies, and I think there are only 7 minutes remaining for my time, 6 minutes actually, to be precise. I would like to thank you all for your patience, and I will be happy to answer any questions. Thank you so much.
Thank you, Rahul, for a very interesting lecture on viruses. In fact, all of us, in fact, many of us have seen the viruses only in the books, never, never under the microscope. But it was a wonderful journey that you took us from basics to advanced virology.
My pleasure. Yeah.
So the topic is open for discussion. Dear participants, if you have any query, you can raise your doubts and queries.
Hello, sir.
Yes, ma'am, please.
Yeah, could I, first of all, congratulations to you for such a wonderful work that you have done. And we are very fortunate to get exposed to this nice journey of SARS-CoV-2, because we have been reading so many things in different ways, in different reports, and everyone was so confused. And yeah, we got a lot of insights into the SARS-CoV-2 infections and how actually the research was done in that at that time also, and now also how it is going on.
Yeah.
So wish you very happy new year, and wish you very good luck to continue your work, which is very excellent work, an excellent presentation. So yeah. Now, few questions I want to ask you. Like same with me, many others will also have the same doubts. Um, we were discussing about long COVID, right? So as you said in the beginning, that after 1 year or 2 years, also people might get some problem like related to heart or memory loss and all that. So how long it might, I mean, is there any study related to how long it continues, or it is a lifelong problem, or how it is? And when it was correlated to the animal studies, like how very nicely it was performed in animals. So how it was extrapolated, the results which were got in the animal models, how they were extrapolated to humans.
Yeah, so to answer your first question, how long the symptoms persist for long COVID is we are still tracking and we are still observing even after 2 years after infections. So in animal models, because of the short lifespan of the mice, we were not able to track. We of course saw it until the end of their life, but it shouldn't be the case in humans because our body is very amazing system. It overcomes the problems, but it cannot avoid some long-term impacts. Like we are trying to understand if this gives rise to Alzheimer, dementia in later part of the life. And that's overall goal of my lab, to understand the long-term consequences which viruses can impact our quality of life. Uh, so I would able to answer you for this question maybe a few years from now.
Few years down the line.
Yeah. To answer your second question is how the results were extrapolated for human studies. Yes, so we are currently doing a preclinical studies, and there are some human trials going on for other therapies, which other researchers have worked on. So far, there has been limited success, but what has been observed is the vaccinated animals or vaccinated humans are more secure for long COVID, especially individuals who received vaccine before their first infection. They are more secure compared to the humans who were infected first and then got infected. And we must understand that or consider that the genetics, human genetics has a major role to play in the disease severity, or like let's say the overall impact of the long COVID on their life. So that is more important question. Like my goal is to understand the host factors that contribute to the disease severity. So in future, if a newborn baby, if we can test their host factors, we can immediately understand which disease or which viruses can impact this child in future, and we can vaccinate that child beforehand to improve its quality of life and maybe increase its lifespan in future. So that's my overall goal.
Excellent. And then few more things, like you have tested many drugs for the treatment and efficacy and all that. Remdesivir was very frequently given to many patients.
Yes.
At least here in India. So, can you comment on some, I mean, the effects? And there were so many controversial reports about remdesivir. Some doctors were recommending initially, people were giving malaria this thing, because that time, I mean, the remedy was not known. So malaria medicines were also given, and then remdesivir came into picture.
So remdesivir, to my understanding, it does inhibit the virus because we have used it in the animal models and it has cured the infection to some extent. But again, the response of the drug is dependent on your genetics. So, and that is why clinical trials are recommended, because the drug cannot act similarly on all humans. But the impact of, I think remdesivir was highly useful in the beginning of disease, beginning of infection. So if you are giving in the later phase of infection, it is of no use, because then what we are experiencing is the malfunctioning of our immune system and the disease severity because of immune system and not because of the virus anymore. So the treatment at later part, why remdesivir was not effective. That's what my understanding is. But it was very clear by many scientists that the remdesivir shouldn't have been given for more than like 9 days. But I think it depends on humans. For some humans, it would have still been effective because the virus replication was ongoing for a longer time in those individuals. There have been
Yeah.
so many discrepancies,
Yeah.
mistreatments during
Yes. Yes.
that, and it has impacted my personal life as well. So
Oh,
I
so
don't
sorry
want
to
to
know.
go
Mm.
in detail for that, but yeah, there are many things to comment on.
Yeah, yeah, and there were many side effects also, it seems, for
Yes,
the remdesivir. Yeah,
yes.
yeah. And my third question is, normally, I mean, since you are working on the viruses and
Mhm.
we get vaccinated, I mean, our newborns,
Yeah,
that is a traditional way for many
yeah,
different, I mean,
yeah.
many different inoculations are there. So once we are inoculated for, say, polio or
Mhm.
maybe typhoid or maybe for other, or, I mean, for other viral diseases,
Mhm.
so normally it is assumed that we get
Mm.
immunity lifelong. We frequently
Yes.
never get inoculated for many diseases. I mean, once or
Mhm.
twice or
Mhm.
maybe booster dose.
Yeah.
But in case of SARS-CoV-2, mutations
Mhm.
happen so fast. Like
Yes.
Delta, Beta, Omicron, Washington. There were so many. I mean, the variants were there. So
Yes.
why such fast mutations this virus was showing? And how, because it was very surprising, or it was very new to all of us, that how any virus can mutate so fast, and the vaccine which was given earlier is not effective for the second variant. That
Yeah.
type of things were also there, or I mean,
Yeah.
in your research also you have shown that the severity or the protection that we are getting or the immunity that we are getting may vary if you are vaccinated, not vaccinated, and also all other things.
Yeah.
So, can you comment on these mutations and how is it possible in a virus that so fast it is mutating?
So the entity that replicates fast mutates fast. Mutation is a dynamic phenomena. Virus is not controlling it. Again, going back to Stryer, understanding from Stryer, DNA polymerase 1, DNA polymerase 2,
Yeah.
one has proofreading activity, the other don't.
Mm-hmm.
So the errors made by DNA replication or nucleic acid replication are, and these are RNA viruses, they don't have any proofreading, so that causes more mutations. More and more mutations are acquired by the fastly replicating virus. Like the virus replicates within 12 hours. It can make itself into second copy within 12 hours, and
Mm-hmm.
that gives his ability, that the ability to virus to mutate fast, and especially on different backgrounds, immune backgrounds, the mutations which are favorable. So this is the evolution, right? Like we have evolved with the mutations.
Mm-hmm.
Bacteria or viruses also evolved with their mutations, and their
Mm-hmm.
dynamic phenomena, so they cannot be controlled. But why SARS-CoV-2 is mutating faster, that I don't know. But I know there must be reasons behind it. It may be the polymerases that it is making use RNA polymerases, incurring more and more errors, or it may be that because it transmits from one individuals to other individuals very fast, it gets on new backgrounds very fast, and these new backgrounds provoke newer mutations, collection of newer mutations, and make the virus stronger or weaker. It depends. So I think it will continue. It will continue for this virus and the other viruses too.
Oh,
Yeah.
okay. And then one more thing I wanted to ask you is about the, I mean, we have come across many reports about the gynecological implications of SARS-CoV-2.
Okay.
So, like particularly young girls or women, they
Uh-huh.
also face a lot of problems, like how you said, like long COVID or post-COVID
Uh-huh.
effects.
Okay.
I mean, even after recovery,
Okay.
it continued for quite a long time, and maybe
Okay.
still some ladies are still facing the issue.
Yeah.
So, if anything, I mean, is it related to COVID or?
It may be, but I haven't came across the papers specifically talking about the feminine problems for SARS-CoV-2. But in general, it was observed that male individuals are more susceptible to long COVID. Even females do. But there are so many, the list is vast. Um, so it might be that viral antigens are stuck in other parts of the body, maybe in females more, and they are causing inflammation, and inflammation is resulting in these complications in general. But yeah, I have to look into in this particular aspect of the long COVID, and then I can answer you.
Oh, okay.
Sure.
Yes. Thank you. Yeah. Thank you so much. Thank you.
Sure.
Yeah. Hello, sir.
Hello.
Yes. Yes.
Yeah, this
So
is
can you tell us something about the
Patil.
HMP virus, HMPV?
Okay. The new emerging virus. Yeah, I have also just read about it. I think it is causing respiratory infection like respiratory syncytia virus. Something similar to SARS-CoV-2, but not exactly similar to SARS-CoV-2. Maybe its transmission is low, and the disease severity may also be low, as there are many factors of pathogenicity. So far, to my review of the news, the virus is not dangerous or lethal, but none of the virus infections should be taken lightly. If there has been increment in the cases, we should all wear mask in the public areas to avoid infection by these new variants, new viruses. It can keep us as well as our society safe. Masks are the most important tool to get protection. But the virus will come to our institute. We are the government institute of the United States, and we get access to these variants very quickly. So in future, we may get the hold of this virus, and I will be able to work on it very soon.
Thank you.
Sure.
Yeah, hello, sir.
Hello.
Yeah, I do have one doubt. I just want to confirm, or you just confirm it. After taking the vaccinations in India, right now there are the many cases of the heart attack, sudden heart attack.
Yeah.
The one attack and the person died there on the spot, and no age range. Means
Yeah.
today there was a news that 8-year-old girl died
Oh, okay.
because of the heart attack in her class. So if there were rumors, or there are the rumors that as we have taken that COVID vaccine, we have taken the 2 doses, right?
Yeah.
So is it having any relationship with this heart attack and the COVID vaccine?
To my understanding, until now, it is not related, because the virus needs to replicate in your heart, and then it can damage some basal cells in the heart. Vaccine doesn't cause virus replication, but I don't know if the spike protein is combining with the fibrinogen and it is causing any blood clot formation, and those blood clots are getting trapped in smaller vessels in the heart or brain, which are causing these implications. If it is caused by mRNA vaccine, the vaccine, protein vaccine, attenuated vaccine, that I don't have any idea, but I'm for sure going to study this in detail for the vaccine as well, and see if it is causing any manifestations in the cardiovascular tissues in future. But for now, I don't think it is caused by the vaccine, but I'm not sure.
Thank you, sir.
Anybody's there?
Hello.
Yes.
A very good morning, sir.
Good evening to you.
My name is Priyanka Vadi. I'm currently pursuing my master's degree and I'm in the first year in RC Patel ACS College.
Okay.
I'm astonished and awestruck by the phenomenon and remarkable work you did on the SARS-CoV vaccine. Sorry.
Thank you.
I just wanted to ask you that you said you you searched about how the mice is losing their memory after getting them vaccinated. Sorry, after getting them infected.
Infected, yeah.
Are there any specific tests? How do we know that they are losing their memory?
Yeah, so the behavior test that we did is the test to understand their if they're losing memory. But there are some other tests like fear conditioning test. Am I still sharing the slides? Okay, let me share it again. Yeah. Yeah, here. The can you see the slide?
Yes.
Yeah, so you can see the habituation deficit we studied with the open field test, but there are some tests like Y maze test and fear conditioning test where you give a small shock to the animal. When the animal touch different bars, it gets the shock. Whereas if it learns to avoid the those bars, it is safe. So we can have this kind of behavioral test to understand the associative learning or memory. So there are more studies for understanding the behavioral deficit for short-term or long-term memory, for associative learning or memory, or just the habituation deficit. And I'm currently working on that.
Okay. And my second question is that you said about the long-term
Impacts of SARS-CoV-2. Yeah.
Is it possible if we are vaccinated and still the virus is present in the body and we can't get rid of it?
I would say if in 10% of individuals who are infected before vaccination, the antigen may be present in the body. They haven't reports that even after 1 year, they were able to retrieve the spike protein from the blood. So there is a possibility if you are vaccinated after infection.
Okay. Thank you, sir.
Sure.
Sachin Patil, sorry, Sandeep Patil, sir. Can I request you something? Are these lectures recorded?
Yes, we are recording the lectures.
Sir, and will they be shared later?
Sure, ma'am. I'll share with all the participants.
Yes, thank you. And the contact of the speakers.
Sure. Yes.
Yeah, thank you, sir. Thank you so much.
Yeah, but I also want to tell you all that there is some unpublished data in the presentation. So keep it to yourself until it gets published.
Sure.
Thank you.
We'll take care of it. We'll take care of it.
Yes.
Yeah.
Sure, sir.
So Rahul, last question.
Yeah.
There are many students in the group.
Yes.
So you just tell them that what it actually takes, what it really takes, from RC Patel to NIH.
Okay. I would say what it took me to go from RC Patel.
What we could get from your case is that it takes only sincerity and honest efforts. That is my reading. Reading as far as you are concerned.
Yeah.
So you tell the students,
Yeah.
what exactly we need, for this fascinating journey.
I can talk separately on this, but just to tell you in short, I am very common. I'm a student with very low IQ. I would say I have secured only 53% mark percents in my 12th. I have gone through ATKT in my bachelor's degree, so you can really make it that I'm not very intelligent, but I was always fascinated by research. I remember I did first project with Dr. Ullas Patel sir at RC Patel. And then somehow the term research was very attractive to me. I was like, I want to do this. I don't know if I can or I can't, but I really loved it, and I continued my steps towards it. I didn't care about the failures. I just kept on trying, like the ant which climbs the wall. I I just kept going, and I reached there. And now I am working at the best facility in the world with all the resources. I don't need to write for funding. I get like 6 crores of rupees every year. I get like supporting staff, scientists, uh, postdocs working for me. So it became easy. It looks easy, but it really tests your patience. Um, like at this age, for many students, I would give you a concept. Um, it's your passion. You should follow your passion as if you're following your girlfriend or boyfriend. Like you want to do anything for them, right? Anything. You don't have any restrictions for your time, for your pleasures or anything. You just do anything for it. So if you are in love with your passion, and if you're treating your passion as your girlfriend or boyfriend, you can succeed. You can get it. It's just that you have to persevere, as Dr. Ravi Patel sir said. You have to love it. You have to persevere it. If you don't love it, you will have less energy. If you love it, you will have more energy. And I would also say, don't do procrastination. Um, don't say that, okay, I will begin it from 1st January. I'll begin it from Dashera or Diwali. Don't have these limitations. Start right away for to step into journey of your passion, towards your passion, and I think you will get it.
So, student friends, message is very clear. I guess all of you have got it, and we expect many Rahuls from you also. So with this, I thank Rahul for joining us all the way from US, accepting your invitation and delivering such a wonderful lecture, inspiring words and lots of things. Thank you once again, Rahul, and
It was indeed my pleasure. Thank you, Dr. Ravi Patel sir and Dr. Sandeep Patel sir, for giving me opportunity to talk, and I will be available for any guidance for the students.
We keep you troubling in near future also, Rahul.
Yeah, no issues. I think it's my duty. So
thank you, Rahul.
Thank you.
So we'll end this meeting now, and once again, thank you and good day, Rahul.
Thank you. Good night to all of you.
Good day.
End करू ना?
End meeting.